Hello, everyone. Good morning or afternoon. We are going to get started on our second part for the full length. This would be involving the not so peasy questions and less peasy questions that I put in the Google form attached in Sakai. All right, so I'm going to turn off the camera simply because it's not needed, but okay. All right, so let's start the screen share. So definitely check out the Google Doc that I put on Sakai of how I'm going to be doing the full length area analysis. So I split things up into either easy peasy or less peasy or not peasy. And this PowerPoint, the second one, PowerPoint part 2, is going to be discussing the less peasy and the non peasy questions. All right, so I'm going to be sharing my screen and showing you guys work. All right, so we can start off with question 12 from ChemPhys. Oh, okay. Share. All right, hopefully you guys can be seeing both the PowerPoint and the TPR. Okay. So start off with question 12. Okay, whenever it is, I just refresh it. Right. Yep. What you want to see is a positive trend, 493, 496 to 502. And then I just kind of didn't increase. That's what happens when you don't area analyze. You just keep taking the test over without studying. It's not good. All right, question 12. Okay, it's a freestanding question. All right, it asks, which of the following statements regarding RNA molecules is not true? Basically, for this, you just have to do the process of elimination. When it says not true, it means that the other options that are the wrong options are true. All right, so we're just looking, typically, if depending on the difficulty of the question, it'd be easier to like sometimes just choose the one that's straight up not true. But if you're having troubles, then definitely use a process of elimination. Okay, first thing, A, RNAs can act as an enzymes to catalyze reactions. Yes. If you know the term ribozyme, here I have in the content, you know that RNA can also act as enzymes, not just like as messenger RNA or rRNA or tRNA. It can catalyze different reactions, like for example, the uh, the enzyme telomerase is a ribozyme. Okay, so we can cross that one out because it's true. All right, so at the time, I didn't know that some RNAs have more than 4 different types of bases. So now I thought it was like the classic uracil, adenine, guanine, and cytosine. But there's actually different ones. So here's the classics: adenine, guanine, cytosine, uracil. But you can have some modifications. You can make a methyl adenine by adding a methyl group to the nitrogen on adenine. Or you can add a methyl to guanine or a methyl to cytosine. Or maybe I just have a different type of uracil in which you have a new amino group right here. Rather than having 2 amino groups, you have a third. These modified amino acids typically are found in the tRNA. So tRNA is the transfer RNA, or the RNA that kind of moves the amino acid to go sort of meet the codon in the ribosome. And once it meets the codon, the anti-codon on the tRNA is going to match it, and then you have your amino acid there present, perfect. That structure is kind of looks like a loop. Like if you were to draw a tRNA, it's kind of like this, and then here you have your amino acid, and then you have the anti-codon here, and then you have the codon on the ribosome over here, and then these guys match, and then there's just different bases involved in, like, let's say there's like a different base here, modified base here, modified base there. The tRNA has modified bases, so B would actually be correct, something I didn't know at the time. Okay, some RNAs are synthesized in the nucleolus. Yes, the rRNA. So just to remind you guys, this is the cell, this is the nucleus, and then the nucleolus would be the dark spot in the middle of the nucleus. That's where the rRNA is constructed. It's synthesized. That's the ribosomal RNA, the things that make up the ribosome. Okay, so the last thing must be false, right? If everything else is true, then this must be false. Our RNAs are insusceptible to alkaline hydrolysis. Well, this is false because something you might remember is that our RNA is less stable than DNA, right? DNA is the thing that stores our genetic information. RNA is kind of the thing that the intermediate in the central dogma between DNA and protein. So why is RNA kind of less stable than DNA? Well, what's the difference between DNA and RNA? Well, the difference is that they have a in the RNA, you have a hydroxyl group here because this is a ribose sugar, and in ribose sugars the carbon groups have an OH attached to them. However, in DNA, this OH here, like this is the first carbon right here, is second carbon, third carbon, fourth carbon, fifth carbon, or ribose is five carbons. Here in the second carbon, for DNA, this OH group is not here. However, in the RNA, it is here. And recognize the name, deoxy, right? That's the name of DNA, deoxynucleic acid. So there's a deoxy, means a missing oxygen, right? So, and RNA is just ribose nucleic acid. So it's just a regular ribose, but in DNA, it's deoxynucleic acid, right? There's a missing OH group. So what this OH group does is that it's susceptible to alkaline hydrolysis. So alkaline means base, and hydrolysis means to like break up. Okay, so what's going on here? Okay, here we have an OH group. The OH group snatches the hydrogen from the alcohol group right here. As you might know, like from the structure of RNA or DNA, the third carbon and the fifth carbon of the of a nucleotide, like let's say this is one nucleotide, this is another nucleotide, the phosphate group is attached to the third carbon and the fifth carbon, and this is your phosphate group, right? It's the phosphate sugar backbone. It's like the phosphate group, the sugar backbone right here, and then you have the base, right? Whether it be adenine, uracil, guanine, adenine. I'm not sure if I said that one already, or cytosine. But yeah, this is your backbone here. And what happens is that when this base takes away this hydrogen, what happens is the oxygen now becomes very nucleophilic, right? I mean, it's nucleophilic to begin with because it has lone pairs. However, now it's even more nucleophilic. Why? Because it has a negative charge, right? The hydrogen was snatched away by that base. So what's going to happen is this nucleophilic oxygen is going to attack this phosphate group. And what's going to happen is the phosphate is like, okay, there's way too many bonds on me right now. I'm going to take off one of the bonds. And one of the bonds happens to be that bond to the fifth carbon, right? This fifth carbon has a hydroxyl group here initially, but then the phosphate group takes away the hydrogen and bonds to the oxygen. So what happens now is just the oxygen gets the pair that it shares with the phosphate group. This is a pair of electrons being shared. That's what a covalent bond is. The phosphate rolls off and the pair goes back to the oxygen. What happens is that now it just splits off, right? The once dinucleotide has now become two nucleotides, two mononucleotides, right? The phosphate got off of this one, it went on to this one. Now this base is doing its own thing, and this base has been left behind, right? That's why RNA is unstable, because it can just break up like that due to alkaline hydrolysis. And you might be wondering, hey, where did that hydrogen come from? Like I thought the oxygen was kind of like ripped off and now it's by itself. Well, in the alkaline hydrolysis, what can happen is that the base, this base right here, when it took that hydrogen, it became H2O. The H2O, this one of the hydrogens from H2O could have been snatched by this oxygen group, making an alcohol group. Okay, that's just some details on the mechanism. Do you need to know all that? Like, do you have to have that memorized? No, but it's good for understanding, right? It's most of the information I'm going to be giving to you guys is more for context rather than like memorization. All right. Okay, so now let's go on to question 25. Okay, there we go. Remember, guys, whenever I read a question, whenever I do a section, I read the question first to see if I need to like use the passage to actually answer it. Generally, for the less peasy and the non-peasy questions, they're really tied to the passage. So typically, we are going to have to use them. But okay, let's see here. It says, which of the following can the researchers conclude strictly from the data shown in Figure 2? Okay, so let's look at Figure 2. This is Figure 3. Don't need this one. Figure 2 here. Okay. In my first glance at when I reach this passage, the first thing I want to do is like look at the big figures, right? Just to memorize their locations and what they're talking about. This is a mechanism. Cool. This is a figure determining the rate of DOPC hydrolysis. I don't know what DOPC hydrolysis is yet because I didn't read about it. But I'm just going to note that the y-axis has percent mole, which means concentration. And over here we have time. So typically whenever we have time and concentration, we're going to have some sort of process, right? Some sort of thing that creates a product or uses up a reaction. And for that reason, in this case, we're creating a product, right? We're creating lyso-Pc because the percentage of lyso-Pc increases with time, right? Time increases in this direction, lyso-Pc increases in that direction. It means with increasing time, there's increased mole percent of lyso-Pc. Okay, so that's what I know so far. Also, I'm just looking at the line here. And what we got at the line here is it's a straight line. If we have a straight line, there's no curves. That means that there's no change in rate, right? So well, what's the rate? So for example, rate is like miles per hour. That's a rate, right? Two different things, like in this case, concentration over time. We can derive differences in rate by looking at the line, like the slope of the line. Here, the slope of the line doesn't change, right? Like let's say over here, we had this line, right? This is a straight line. The slope doesn't change. The rate isn't changing. However, if we were to have something like this, well, here the slope of the line changes, right? Here, if you were to take the rise over the run, remember how to do y equals mx plus b, the slope changes constantly. And for that reason, that means that the rate is also changing constantly. And in this case, the rate is exponentially growing, right? It's growing in this direction. If we had something like this, that means that exponentially, like if we had like a graph like that, and the rate was like this, right? That means that you're decaying, you're decreasing your rate exponentially. And here I just have some graphs to like explain kind of like how to interpret kind of rate and all that. So like here, for example, I'm just going to break this down real quick. You know, if we were talking about like a vehicle on a street, right? So let's say we have a stationary object. The vehicle is not moving from its parking spot. So with time, as time increases, its position isn't going to change, right? Let's say that this is X, right? Position. It's just going to stay at the position right here. And with time, it doesn't move. So it's going to be straight. Okay. So what can we derive from that, right? Well, what can we derive is with each additional graph. So like the graphs go in this direction. With this graph, we see what's it called? The distance over time, like this. So let's split this up real quick. In this graph over here, we see distance x time. Over here, we see velocity x time. And here we have acceleration x time. I'm sorry that my handwriting is bad, but you just need to like hear the concept rather than write it down. So here, the distance is, you know, changing with, in this case, in this graph right here, distance doesn't change with time. And if there's no change in distance or time, is there any velocity? No. Okay, if velocity is 0, then that means there's no change in velocity. So acceleration is also going to be 0. But let's say we have uniform motion, right? Let's just say that the car is creeping towards like it's going, let's say 40 miles an hour consistently, right? So it's going 40 miles an hour consistently. That means that in 1 hour, it's going to, what's it called? Like let's say this is 1 hour right here, right? If it's going 40 miles consistently, say it's 1 hour, 2 hours, and then in 1 hour, it'll reach 40 miles. In 2 hours, it'll reach 80 miles, right? That's a consistent uniform motion. So what's the velocity going to tell us? Well, the velocity really hasn't changed based off of our position graph. So the velocity is going to stay consistently at 40 miles an hour, right? So this is velocity now on the y-axis, rather than over here, it was just position. But now we have velocity, right? V for velocity, and then A is for acceleration. And so velocity stays consistent at 40 miles an hour. Let's say over here, it would be 80 miles an hour. But we never do that with uniform acceleration, right? I mean, sorry, uniform motion. It's just a consistent velocity. So it stays like this, even with time passing. Remember, we had like 1 hour, 2 hour, the velocity hasn't changed at all. So the velocity is just going to stay here. We have a straight line. In this case, if there's no change in velocity, we have no change in acceleration. So do you see how that correlates with this graph over here, right? Like how you can learn something from this graph. Basically, with increased time, there's also a steady increase in lyso-Pc. And the line is straight, which means that the rate of the reaction is constant. Let's see over here, right? We have x and t. here we have that curve where we're talking about, right? The exponential increase, right? So over here we had that in 1 hour we'd reach like 40 minutes, but over here in 2 hours, instead of reaching, sorry, not 40 minutes, 40 miles, over here we can like reach 90 miles, right? The position has changed, right? It takes less time to reach this height. Like over here, you know, like the line drawn over here, say like over here, the line drawn, like let's actually draw it out, right? In 2 hours, we hit 80 miles. In this case, in 2 miles, sorry, not 2 miles, in 2 hours, we don't even know when we hit 80 miles actually over here. You see how it takes less time to hit 80 miles, which means that our velocity is changing. And how is it changing? Well, if you use y equals mx plus b, you'll see that the velocity, what's it called? It's no longer one single velocity, right? If this is velocity on the x-axis, it's no longer a single velocity. If it was a single velocity, like here, it'd just be a straight line. But the velocity is creeping up. Like let's say we had 1 mile per hour, and then 2 miles per hour, and 3 miles per hour. The velocity is changing. And if we have a change in velocity, then we have acceleration. And the reason why acceleration is a straight line is because it's constantly accelerating at the same. It's constantly accelerating at the same amount. Like let's say you're constantly accelerating by 1 mile per hour. Okay, cool. That means you're going to go from 1 mile per hour to 2 miles per hour to 3 miles per hour. But those intervals are by 1 mile per hour. So acceleration is going to be 1 mile per hour, right? Okay. So that's something you might have talked about with Justin in terms of projectile motion and all that, but I just wanted to go over it just so you can break down this graph, right? So you should always be looking at the y-axis, the x-axis, what the slope of your line is. Is there a linear? Is it linear or is it exponential, right? Okay, let's actually get to the question. Okay, so the question says, okay, right, what can be said about figure 2? All right, so here we also have a legend, right? This is called a legend. It tells you information about the graph. Here we have DOPC plus 5 moles of raclopride is the square. Okay, we have the squares over here. It looks like squares are constantly increasing. These are error bars, right? Just in case like they got the data wrong. They're like, okay, it might be here or here. Right? This is just error bars. And then when you have DOPC only, so it seems like we have an experiment, right? Typically with experiments, you have a control group and then an experimental group. And here we have DOPC only, and here we have DOPC plus raclopride. So this is probably your experimental group, and this is your control group, right? When you're not changing anything, you just have DOPC there. I don't know what DOPC is yet, but that's what I can derive from just looking at the graph itself. And here it says we have circles for DOPC. Where's circles? I don't know. Where is circles? Oh, it's down here. Circles are all the way down here. Okay, so it seems like there's not even a growth of DOPC only, right? There's no, when you have DOPC only, looks like LisoPC doesn't really grow much, right? It's kind of like there. Like over here we have a growth and here we don't. So with time, DOPC only, that condition, LisoPC is not being generated as much as when you have DOPC plus raclopride. Okay, so let's figure out what DOPC actually is, right? So what I'm going to do is I'm not going to read this whole thing, right? I'm going to scan for what DOPC means. So I'm looking. There's nothing here to really like tell me DOPC. Okay, so I'm going to continue forward. Okay, next. Like DOPC is not here in this paragraph, so I'm going to keep going. Okay, using a model system of plasma membrane. Okay, I remember what a plasma membrane is. It's like a bunch of phospholipids. Okay, cool. Consisting of a unsaturated lipid. Okay, unsaturated. That just means that we have a double bond somewhere. Lipid 1,2-dienoyl S whatever, blah, blah, blah. DOPC. Okay, perfect. I found DOPC. Cool. So we have that unsaturated lipid equals DOPC from the plasma membrane. Okay, that's a cool visualization, right? We have a visualization of a plasma membrane. It's a bilayer. And then one of these fatty acids, whatever, is DOPC. Cool. Scientists measured the production of blah, 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 lysopc. Okay, so now we know what lysopc is, right? Our y-axis. In the presence and absence of raclopride. Okay, so there's DOPC and there's also, what's it called? LisoPC, which is being generated. This is the production of this, right? So this is being generated in the presence of raclopride. Okay, so they're trying to see basically what raclopride does to the production of LisoPC, right? That's what it's saying. It's saying production of LisoPC in the presence or absence of raclopride. Okay, cool. So far, I don't have enough context, right? Like I want to know like what the reaction is, right? They said something about a production. Okay, what's the reaction? Okay, here, to investigate the effects of saturated lipids on raclopride-mediated lipid hydrolysis. Oh, lipid hydrolysis. What is DOPC? It's a lipid, right? Okay, so DOPC is going to be hydrolyzed by raclopride, right? It says here, raclopride mediated lipid hydrolysis, which means our DOPC, yeah, the DOPC membrane was doped into saturated lipids across a range of dopant concentrations since membrane fluid. Okay, I don't need to know all this. Who cares? Okay, the doped lipid was used where blah blah blah. DOPC. Okay, and we have a bunch of stuff here, right? Nothing about DOPC, right? All these like acronyms here. Where is that in this figure? So I'm not going to really pay attention to that, right? I just want to focus more on DOPC. Okay, if there's nothing about DOPC, fine. I might have to go up into this thing to like figure out what happens, or maybe the mechanism, right? Okay, so here we have a membrane lipid, and here it says that the proposed reaction mechanism of lipid hydrolysis. Okay, so we know that DOPC is a lipid that becomes hydrolyzed. That's what it says in this first sentence over here. And then I'm guessing this is DOPC. This is the raclopride, right? It says it becomes hydrolyzed with raclopride. So must be DOPC. We know that it's a lipid. Okay, this is definitely a lipid structure, right? This is we're here that the long carbon chain that is a lipid structure. We have the raclopride, and what happens at the end? From DOPC, we have lyso-PC. Oh, okay, cool. So that might have been like that might have seemed unorganized, but really it's a really fast thing, right? All you have to do, okay, find DOPC. Okay, cool. This is what DOPC is. Can I have enough information to derive information from this graph based off my understandings of DOPC? I can't. Okay, let's find a little bit more stuff. This stuff doesn't deal with DOPC. Okay, I'm gonna backtrack now. And bam, I found whatever I needed, right? So you don't have to necessarily read the entire thing because like this is just one example, right? And some other examples, this single paragraph here tells you all you need to know. So you won't need to know this, right? So it's just all about strategy, becoming efficient with your short-term memory, right? Okay, so now we know that when you have DOPC, right, your lipid, and there's no raclopride, basically the DOPC just exists, right? It's a plasma membrane lipid, so it's just there. It's not going to be hydrolyzed, seemingly, right? Because most of our stuff is here at 0. It's not going to be hydrolyzed by something and turned into lyso-PC, right? Lyso-PC is the product after the hydrolysis of the membrane lipid, aka DOPC, right? Okay, but when we have DOPC, the membrane lipid, and we have raclopride, what happens? We have a bunch of lyso-PC, which means that DOPC is turning into lyso-PC consistently. Okay, that's what I know now. Okay, now that we've objectively figured out the graph, let's look at the options. All right, so which of the following can the researchers conclude strictly about the data shown in figure 2? All right, let's do the process of elimination real quick. Okay, so it says here, lipid hydrolysis cannot occur in the absence of raclopride. What's lipid hydrolysis? That's the production of lipo-PC, right? Right here. Lyso-PC. Where does lyso-PC become generated? By the hydrolysis of DOPC. So let's revisit that answer choice. Lipid hydrolysis, okay, so DOPC turning to lyso-PC cannot occur in the absence of raclopride. Okay, let's look at that. So here we have DOPC with raclopride. Okay, we have lyso-PC being generated. Awesome. When we don't have any raclopride, seemingly there is nothing being generated, right? However, look at the language being used here in this question stem and then this answer choice. It says here, which of the following can the researchers conclude strictly from the data shown in this figure? Okay, cool. So only in this figure, right? And look at the language used in this answer choice. This lipid hydrolysis cannot occur in the absence of raclopride. It's like, doesn't that seem extreme to you? It's like lipid hydrolysis never, ever, 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 ever can occur without raclopride. This random molecule that they're testing out in this experiment. And if you remember from our previous kind of run at this question about raclopride and what it is, right? Remember we talked about how it's a Bronsted-Lowry acid because of this nitrogen right here. Well, if all you need to have for lipid hydrolysis is a Bronsted acid, there's so many Bronsted acids out there. Raclopride isn't the only Bronsted-Lowry acid. So it's just like for this kind of thing to be correct, it's too extreme. And that's something that I didn't know before, right? That in the sciences, just like in cars, you can have things that are too extreme. For example, they talk about, let's say, the theory of gravity. And it's just like, oh, well, things, because of gravity, things will never go up, right? Things will always go down because of gravity. Well, we know that's not the case, right? You can put force into something and cause it to move up, right? So this thing over here, it's too much, right? It's saying here, lipid hydrolysis cannot occur in the absence of raclopride. Here, sure, in this case right here, we don't have much going on with lyso-PC creation. However, that doesn't mean if we were to introduce a different, what's it called, kind of bronzolaric acid, instead of here 5% mole of raclopride, we put 5% mole of something else, then we can have lyso-PC generation. But just strictly from this graph right here, we can't say that no matter what, raclopride is the only thing that can cause this kind of lyso-PC to be created, right? So this is too extreme. Okay, let's keep looking. So DOPC plus 10 mole percent of raclopride would have a higher rate of lipid hydrolysis than DOPC with 5 mole raclopride. Okay, can we know that? I mean, based off the strictly, so remember in the answers, this is in the question sum, it says strictly from this data shown, can we know that adding more percentage, like having a higher concentration of raclopride would increase this? Maybe, I mean, who knows? But this graph does not tell us that, right? This graph only tells us what? This graph tells us at 5% mole raclopride, we have an increase. And then when we have no raclopride, we have very small, if zero significant DOPC being turned into lyso-PC. But it doesn't tell us, oh, if we increase the concentration of raclopride, then it will be way more production of lyso-PC. I mean, think about it, right? What is raclopride? It's a bronzolaric acid. If we have a bunch of acid in a solution, is that going to is that going to help us? Like, I'm sure it might help us at a small percentage, but if there's too much acid, right, then you might have some unnecessary reactions occurring. So based off the graph itself, we can't say that 10% would be a better hydrolysis for DOPC than 5%. Why? Because 10% is mentioned nowhere in this graph. It only mentions 5%. We can't jump that far. So let's leave it out. Okay, let's look at D. The rate of lipid hydrolysis with DOPC plus 5% mole of raclopride increases linearly with time. Okay, well, let's look at our graph, right? It says increases linear with time. Does it really increase linear with time? No. Why? Because the rate is stagnant, right? Here we have the same graph, right here. This graph is the same thing as this, right? Just a diagonal line. What is the rate telling you? The rate is telling you that it's staying stagnant, right? It's going straight. Is that an increase? Is that a decrease? No, it's just right here constantly at this velocity. And that's what the slope of this line tells you, right? The rate is consistent. So here it says the rate of lipid hydrolysis increases. No, an increase in rate shows acceleration. Here, we don't have anything telling us about the acceleration, right? We have a straight line. If we had a linear line like this, then okay, we might have some acceleration, right? But we have a straight line, which means that the rate of the process stays consistent. We can cross out D. All right, C, the correct answer. Lipid hydrolysis with raclopride is significantly faster than any background degradation. Okay, what does background degradation mean? Well, it just means that there's degradation happening by itself, right? Just background degradation, something that happens on its own. Well, let's look at it. Lipid hydrolysis with raclopride is significantly faster. Here's the process with raclopride. Here's the process without raclopride, right? Background stuff. And yeah, we can definitely say, okay, this happens way more faster than this happens because this is literally close to zero. I'm not sure if it touches zero or not, but this one is way faster. Like, look at this rate. It's moles over time, and we do have an increase. So based off the graph itself, we can say that it is significantly faster, right? Because here we have something that's close to zero, and here's something we don't, right? So that is a significant increase, and C would be the correct answer. Okay, so now let's move on. Next question. And we have question 32. Okay. Question 32. Let's see it. There you go. All right. So let's look at the question first to see if we need to know the passage or not. Okay, here. It says, what changes in cytosolic conditions would most directly disrupt the catalytic function of the CTA1 glutamic acid 23 and glutamic acid 57 residues? Okay, really quick, let's break down what this question is asking us, right? There's a lot in this question, so let's break down the kind of the terminology found in this question. Okay, what change in the cytosolic conditions? So what is cytosolic conditions? Well, cytosol, that's just the cytoplasm, right? So that just means the environment of the cell, right? The cell is encased in a membrane, and kind of like the kind of like surround the environment of the cell. Like, let's say this is a nucleus, you have ribosomes, whatever. This kind of like medium right here, like this water, that's the cytoplasm. Okay, so it says here, what change in cytosolic conditions would excuse me. So what change in cytosolic conditions, okay, the environment, would most directly disrupt the catalytic functions of CAT1 GLUT23 or GLU57 residues. Okay, so we know that catalytic function, that means that an enzyme is involved, right? A catalyst means that there's an enzyme. And GLU23 and GLU57, that just tells us about amino acids at position 23, at position 57. So whenever you see, that's why you need to know the abbreviations of the amino acids, right? It says the amino acid and then the location of the amino acid. This amino acid is located on the number 20. That's the number, is the 23rd amino acid. This GLU is the 57th amino acid, right? And what is another term for amino acid? A residue. Okay, so it's telling us what conditions in the cytoplasm can cause and change in the residues of glutamic acid and glutamic acid found at 23 and 57. And just this, I'm guessing this must be the enzyme, right? Because there's nothing else to give us any context of what it is, right? It's a catalytic function. There must be an enzyme. This one must be the enzyme. Okay, seems like this one's really kind of intricate with the passage. So we're going to have to read the passage. But mainly, we're going to be looking for these three things, right? You're not going to be looking for anything else. Okay, let's look at it. All right. So blah, 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 toxin. Okay, who cares? Blah, blah, blah. Okay, CTA1. Perfect. All right. So it seems like this is actually going to have to matter. So let's read it, right? Because right before knowing where CTA1 was, like what if CTA1 was down here, right? What if it was introduced down here? That would mean that all this stuff would be nonsense, right? I don't really need to know it. But since CTA is here and we want to have proper context, we are going to have to read it. Sometimes you don't. But in this case, we are. All right. Cholerotoxin is an oligomeric protein complex secreted by Vibrio cholera. Is that relevant? Well, let's think about it. Right now, we are in ChemPhys. Vibrio cholera is a bacteria. That sounds like it would be more related for biochem, right? So in terms of this biology information, do we have to memorize it? No, right? Who cares about this stuff, right? This is ChemPhys. You're looking for organic chemistry, chemistry, or physics stuff. You're not looking for biology stuff, right? Okay, let's continue. The toxin is composed of an A subunit and 5 identical B subunits. Okay, so I'm thinking about that in my head, right? One subunit and six, and so that means one subunit here, 5 subunits here, means 6 total subunits. Okay, the 5 B subunits form a 5-membered ring. Okay, so they're all in a single group. Cool, I don't have to take account into all the different B subunits, right? They're just one big ring. While the A subunit is composed of 2 domains. Oh, what's a domain real quick for a protein? So a subunit in this case would be a section of the protein. So this is a protein. This is the alpha. This is the A subunit. And within this A subunit, you can have domains, right? This domain does something else, and then this domain would do something. Oh, that's kind of not obvious. And then this domain would be doing something else, right? There's domains within the subunit. And what do domains do? They do different functions. Okay. So you have the A1 and the A2 domains. Okay, in the A1 domain, oh, okay, finally, now I know what CTA1 is, right? It's the first A domain, right? So it says CT. What does CT mean? Oh, probably means cholera toxin, right? Okay, cholera toxin A1. Just remember the names are typically named properly, right? So cholera toxin A1 domain, right? That's what this enzyme is, right? The A1 domain. Chlorotoxin A1 domain acts to ADP ribosilate. Okay, so it's going to be adding some ADP to something, right? It acts to do, like, this is a verb right here. They're saying this as a verb. It acts to ADP ribosilate. So they're adding this thing. Ribosilate means to add this ribose thing. So ADP, it is a ribose, right? Here's a ribose. Here's an ADP, or this is actually NADH, but like right here, this is what an ADP, an adenine diphosphate, would look like, right? So you have an adenine, you have 2 phosphate groups, and you have your adenine, you have your ribose, and you have your 2 phosphates, right? That's what an ADP would look like. And I'm guessing here in the NADH, they're just going to split this thing off. Yep, and they do. Here you have your ADP, and then it's going to do things. Okay, cool. Acts to ADP ribosilate and permanently activate. Okay, permanently sounds, it's a strong word, so I'm going to take note of that. And then it says activate GTP binding proteins. Okay, so that means that this A1 domain is going to add ADP to a GTP binding domain to activate it. That's a lot of information to take down, so I would suggest writing it down in a simple form for you guys. For example, so what I would do is I would say CTA1, it says acts to ADP ribosilate, so I would do this ADP. So it's kind of like it's adding, so plus ADP, and then it's going to do that to, actually, so I would form this differently, right? So just to be as efficient as possible, right? So this is an enzyme, right? What an enzyme does is it's part of a reaction. So I'd actually put so CTA1 and then plus ADP to what? The GTP domain, right? GTP binding domain. And it says here GTP binding proteins of G proteins. So I'll put GTP G protein. And then after this reaction right here with the CTA1 enzyme and the ADP, we're going to have G protein or GTP binding protein, whatever, the same thing, right? They say GTP binding proteins, aka G proteins. So G protein active. That's how I'd write that down, right? It might take a second, maybe like 30 seconds, but okay, imagine like reading that over and reading it over and reading it over, right? That can take more than 30 seconds. But taking 30 seconds to just kind of write it out. So an enzyme, have it on the reaction line, whatever the enzyme does, have it on the bottom, have the reactant and the product on either side, right? The reactants here, the products over here. GTP goes from GTP to active GTP, aka G protein, with CTA domain plus ADP, right? That's our understanding of it. Take 30 seconds, write that down. Actively blah, 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 present on the cytosolic face of the cell membranes. Okay, remember what a cell membrane is. This is a cell membrane. This is the extracellular space outside of the cell. Inside of the cell is what? The cytoplasm, right? So it means the cytosolic face. The cytosolic face just means this area right here, right? The extracellular space would be, it would be this area. This would be the extracellular space. The inside of that would be the cytosolic face, okay? Okay, so membranes by interfering with their intrinsic GTPase activity. Okay, so now we know why it turns it on, right? Why does it turn it on? Why is it permanently active? Because it interferes with GTPase activity. So I'm going to add that to my diagram, okay? So it says GTP protein at the GTPase site. This happens, and then we have G protein active, right? So it's not only just the G protein. I mean, of course, it's a G protein, but is the G protein where? At the GTPase site, right? It interferes with the GTPase activity. Awesome. Okay, cool. The CT1 domain uses NAD plus as a resource of ADP ribose. Of course, as we said, the NAD, the nicotinamide it's a long word. I forgot the name for NAD. Well, this is NAD plus right here, right? So what's going to happen is that CT1 is going to use NAD plus, going to chop off this area right here, so we can have our ADP, or adenine, right here is adenine diphosphate. So CT1 domain uses NAD plus as a source of ADP ribose and depends on two critical catalytic residues, this guy and this guy. Awesome. So we have enough information to tackle our question, right? It says here, what is something that can cause the functions of CTA1? As we know now, it's catalytic, it's cholerotoxin A domain, A subunit 1 domain, and the residues that it has attached to it, right? So we just have to influence these guys. What do these guys do? So we're gonna have to look at the mechanism, right? And guys, remember, if this stuff doesn't come like it doesn't come naturally, look, I fucking got this shit wrong, right? Like if doing all this stuff takes a lot of time, and it does, what's the strategy in the BSM? What does it tell you? It fucking tells you to skip this thing, right? Like, this is only worth 1 point, and look at all the effort that we're putting into it. If you're getting stuck and there's easier questions to get to those first, and then have this going later. And that's why the strategy that I tell you guys is go through the entire section, find all the freestanding questions, right? Even the freestanding questions that are found like in passages, like even the passage questions, some of them can be answered without the passage or minimal information from the passage. Get to those first, because you don't want to regret not taking those. Like, you don't want to regret not being able to touch those. Like, that would suck, like, just to not have the opportunity to tackle something easier, right? After you finish all of those, then go to these hard ones, right? Because why? Because each question gives you 1 point, right? So look at all this effort that we're putting into this single question, right? Leave it towards the end if you have time. You just need to be as efficient as possible, right? Like, you don't have to pat yourself on the back for getting a hard question. If you're missing, like, if that time that it took you to answer that one hard question could have been the same amount of time you could have got to answer 3 easy questions, right? Okay, so here we have our mechanism. We have our NAD plus. We have our GTP binding protein. Oh, where was the GTP binding protein again? The G protein? It was on the cytosolic side of your membrane, right? So this GTP binding protein is basically a plasma membrane. You can imagine a plasma membrane over here, and this thing is just poking out, right? Okay, now we have our enzyme CTA1, and we have our just maybe two enzymes needed. Okay, two enzymes are needed. CTA1 is over here. CTA1, actually no, sorry, I said that wrong. Not two enzymes, just two different sections of the enzyme, right? So if you can imagine over here, like this is one section, this is one section. You can imagine this huge kind of thing over here. This huge thing attached to it. We have GLUT23 on the 23 position. We have GLUT57 on a different position, right? So it's not two different enzymes. I said that wrong. Sorry. It's just a huge thing. They're just not drawing the enzyme, right? Okay, so let's see what GLUT23 does, right? So what does it do? Well, it takes off one of the hydrogens from this amino group, and then the hydrogens that is basically snatched by GLUT23. Well, what does that mean? That means that this bond right here, this shared pair of electrons between the nitrogen and hydrogen, well, what is that bond going to do now, right? Because the hydrogen left. Well, what it's going to do is it's going to tackle the ribosome and attach to it. Okay, cool. That's what GLUT23 does. It uses its negatively charged oxygen to snatch a hydrogen so that the ADP can attach to the GTP binding protein and do what? Oh, make sure that's permanently activated. Okay, so that's the job of GLUT23. What is the job of GLUT57? Well, over here, we see closely, we have a dashed line. Okay, what does that dashed line mean? Right? It's not a line like this. They're different. A dashed line means you have hydrogen bonding, right? It means that you have an intermolecular force, right? Intermolecular force. In this case, right here, we have hydrogen bonding. So why is that relevant? What is the point of that? Well, imagine you're an enzyme and you have your substrate, aka in this case the ADP. It's in your active site, right? Well, do you want the substrate to leave? No. So you want to hold it in place. So how are you going to hold it in place? Sometimes you covalently bond to it, right, just to hold it in place, and other times you use intermolecular forces to keep it there. In this case, we have a hydrogen bonding, and look at this GLUT57, right? It doesn't have a hydrogen on this carboxyl group. It has the negatively charged oxygen, just like GLUT23, which means that this oxygen has a negative charge. That means the hydrogen bonding occurring here is going to be super strong, like more than normal. Why? Because the hydrogen bond uh, 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 acceptor group right here with the lone pair is super duper negatively charged. So this hydrogen with the dipole moment that it has, because the oxygen is moving the electron clouds towards itself, making it negatively charged, and this thing positively charged. This positive charge on the hydrogen is going to be super attracted to this negative charge on the oxygen. So it's holding it there, right? Okay. So how can we stop this from happening? How can we influence the cytosolic environment to stop this from happening? Well, let's look at what's necessary, right? What's necessary here is this negatively charged oxygen taking one of the hydrogens from the GTP binding protein. In this case, we have the negatively charged oxygen, what's it called? You have a negative charge over here, making like a kind of super hydrogen bonding. So how can we stop that? Well, if we introduce a hydrogen here and a hydrogen here, kind of those two functions would stop from happening, right? Okay, so how can we do that? How can we take away this ability to take away a hydrogen? Well, we can introduce a hydrogen here, so it doesn't have the ability to take away a hydrogen, or it doesn't have the ability to super hydrogen bond. Well, let's look at our options. The options tells us that we can increase the pH. Well, remember, if you have an increase in the pH, if the environment is more basic than, let's say, our acid here, like this is glutamic acid, or this is a carboxylic acid. If the environment is more basic than the acid, well, then that acid is going to donate its protons super readily, right? Because the acid is telling itself, hey, I'm way more acidic than the environment, which means I'm going to donate my proton. Who cares, right? But if you decrease the pH, right, if the environment is more acidic than the acid, then the acid is going to say what? The acid is like, hey, I'm more acidic. I'm going to donate my proton. And the environment is going to be like, psych, I'm actually more acidic. You're going to have to hold on to that proton, man. You're going to have to hold on to that. You can't donate it, right? So if we were to decrease the pH, then these carboxylic acid groups on the GLUT23 and the GLUT57, they're going to have hydrogens. And when they have hydrogens, you can no longer snatch the hydrogen on the nitrogen on the GTP binding protein. If you can't do that, then this area right here is not going to bind to our substrate. It's not going to bind to the ADP. And if it can't bind to the ADP, guess what? This enzyme CTA1 is not doing its job. So that would be the correct answer. Everything else, like we already talked about this one here, typically for enzymes, if you increase the temperature, you kind of denature the enzyme. So that could have been something, right? But it says here specifically GLUT23 and GLUT57. So sure, increasing the temperature might have influenced the enzyme activity, but it wouldn't necessarily influence these specific activities here, right? So you need something that encompasses all three. That would be the decrease in pH. And then the decrease in temperature doesn't denature the enzyme. It just makes the enzyme work slower. So if the rate of the enzyme was 100 products per minute, if you decrease the temperature, it'd be like maybe 80 products per minute, right? Because it's a colder environment. Okay, so that is the answer for this question. All right. Remember, guys, if you have any questions, definitely feel free to reach out to me. All right, so that's 32. Let's move on to the next one. Here we have 33. Okay, cool. Let's do it. Oh, the same passage. All right, let's see what we got. So the result of the experiment suggests that, okay, so not a lot of information to go off of. So let's find what this experiment is. Typically, for passages, they kind of like give you background information. And then what they do is they say, okay, with this background information, we're going to do an experiment. So it's like, okay, cool. So what's the experiment going to be in this case, right? So typically the experiment where you can find the word experiment is typically going to be towards the end of your passage, right? Okay, so let's see here. An experiment was performed to consider the effect of a mutation in the gene of encoding RF1. Wait a second, I don't even know what RF1 is. So screw it, I'm gonna have to go up here and read for context, right? Sometimes you don't have to do that, sometimes you do. Again, it's being proficient here. It seems like we're gonna have to, so okay, sucks to be us, gonna have to read it. All right, so let's see. Here it says RF1 is a G protein that functions. Oh, it's a gene protein, which means that it's kind of like these G proteins, right? So here we had just like an arbitrary generic G protein. Now we have a name for it, right? The name of the G protein involved in this reaction right here is what? ARF1. Okay, so it's a protein when active as an it functions as an allosteric activator of CTA1. Okay, so basically this G protein here functions to turn this thing on, right? It is an allosteric activator of this CTA1 domain. Okay, cool. So this thing, when active, activates this thing. I would definitely write that down. So let's say ARF1 activates. So I'll put a check mark here for activation. If it deactivated, I'll put ARF1 and I'll put deactivates. It activates, what's it called? CTA1, right? So that's a relationship that we have. Or you can just skip the reaction line. All you would have to do is just put like the check mark here, right? Check, right? This thing checks this thing. Okay, so it activates it. If it were to inhibit it, I'd put the X, right? For the G protein activity, so the G protein, it's saying over here the ARF1, right? Activity depends on the form of guanine to which it is bound. The protein, okay, the protein is active when bound to GTP. Is saying what protein? The G protein. What G protein? ARF1. Always have that in your mind, right? So you don't get anything confused. The protein, aka ARF1, that thing that activates CTA1, is active when bound to GTP and inactive when bound to GDP. Okay, so what does that mean? That means that GTP activates ARF1, which activates CTA1. However, oh, this is supposed to be GTP. GDP, what? Inactivates ARF1. Okay. Which activates blah blah blah. Okay, hydrolysis of GTP to GDP by ARF1 is dependent on the activity of a second separate regulation protein called ARF-GAP1. Okay, so it seems like ARF-GAP1, it shares the same name as ARF here, and over here we have the same name. The only difference is that we have the GAP. What is the GAP probably going to tell us? Like, what does it mean? Well, let's read back, right? Like, okay, so what is, okay, so here you said the GTP inactivates, GTP activates, and then it's activated when bound to GDP. Okay, hydrolysis of GTP to GDP by ARF1 is dependent on the activity of a second separate regulatory protein, ARF-GAP1. So what we can derive is that the GAP, typically you don't really need to know what this means, but it's good to know for context, right? So ARF1, this probably means guanine guanine A phosphate, right? So the A is something else, but that's the way I like to think about it, right? So there's something that deals with the GTP and the GDP is GAP, right? Is guanine something phosphate 1, which I'll probably just A phosphate. Okay, so which activates the hydrolysis action of ARF1 in part by providing a catalytic residue, which stabilizes the GTP binding pocket
of the ARF1 GTP's active site. Oh, okay. So what this thing does, remember this thing is attached to this thing, right? So it says here that the hydrolysis of GTP to GDP by ARF1 is dependent on the activity of a second separate regulatory protein, which activates the hydrolytic action of ARF1 in part by providing a catalytic residue which stabilizes blah, 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 blah. Okay, so what happens here is if we want to write things down, the ARF GAP1 is going to interact with the ARF1 by giving it stability right here. Where is it? Which activates the hydrolytic activity of ARF1 in part by providing a catalytic residue which stabilizes this area. Okay, by stabilizing the GTPase site. Right, so this thing influences ARF1 by what? Stabilizing site. With what, it says? With a residue. What's a residue? That's an amino acid. Stabilize site amino acid of ARF1. Where? At the GTPase site. Okay, cool. We have that written down. You can keep it in your head too. A lot of this stuff is convoluted. So like having kind of like a diagram like this kind of helps you in terms of time, right? If you're trying to be efficient. That's at least that's my advice. Okay, so now we have the context. Let's read about the experiment, right? Okay. An experiment was performed to consider the effect of a mutation in the gene encoding ARF1, which disrupted an alpha helix structure in the GTP binding pocket of ARF1. Okay, so where did we hear about a binding pocket idea? Oh, the binding pocket was when we were talking about this guy, right? This guy stabilized the binding pocket of this guy by adding a residue. But here it's saying that we have a mutation in gene, which means that the residue that was stabilizing it is mutated, which means that there's no longer stability, right? Because this is what the experiment was about, right? The effect of this mutation on the gene of this guy to see what happens, right? Because if you don't have a stable GTP site, well, then guess what? GTP is not going to be hydrolyzed. Okay. So it says the kinetic parameters for both wild type ARF1 and so wild type, all it means is that there was no mutation, right? The gene is nice and dandy, it's chilling. But whenever you have a mutation and you have this thing called delta, what does delta mean? Delta means a change. So what happened? There was a change in residue number 17. That's where the mutation occurred, right? So we have wild type, aka WT, ARF1 protein product, and then the mutant gene. So they're basically comparing the wild type version and the mutation at 17, right? They're comparing those two versions in the presence and absence of ARF1 gap, right? That's the thing that helps things out. And then here we have the results. Okay. So let's look at the legend. It says kinetic parameters of ARF1 GTPase, and then the thing that's not the wild type, so the delta 17 of GTP. Note positive and negative denotes the presence or absence of this guy, right? So this guy, okay, so in this condition, right, we have three experiments, right? This experiment, and this experiment, and here are the results of the experiment. In this condition, this guy is present, okay? In this condition, this guy is absent, right? So we have kind of like a control group right here, right? The control group is this guy, wild type when this guy is not present, and then wild type when this guy is present, and then we have the mutation where this guy is present, right? Let's look at the differences. Okay, so in this case, we have to know what Km and Kcat is, right? So like, what are those two things? Like, how are they relevant? What's going on here? Well, basically, what Km, what it tells you is how kind of connected and how much affinity an enzyme and a substrate have. So kind of imagine like a lock and key, right? If you have a lock and key, what the key is going to open up the lock super easy, right? So you have a Km that is good, right? There's a good connection. If you have a good connection between a substrate and an enzyme, the reaction is going to proceed smoothly, right? And in terms of like, okay, the numbers, basically a lower Km, and you guys will learn the theory in a different video. The lower the Km, the more stronger the affinity, kind of like pH, right? Remember in pH, the lower the number, the stronger the acid. Well, same thing with Km. They can also present it as micro m. What's it called? Yeah. So the lower the Km, the better it is, right? So kind of like the pH. Okay, for kcat, it's the opposite, right? Kcat, it's the higher the kcat, the better it is. Okay, so don't get those two confused. Well, kcat, what it tells you is well, let's say we have enzyme. Let's just draw it out. Enzyme plus substrate, right? And then there's a reaction to get the enzyme substrate complex. So this is K1, right? And then to make the enzyme substrate complex into enzyme plus product, that is your kcat, right? So that's what kcat is. The higher your kcat, it means the higher you're going to make some product. Okay. So now let's look at the results of the experiment. Let's look at the answer choices finally, right? Remember, guys, if you're running out of time, don't tackle these really convoluted questions, right? Because it takes a lot of time and it's only worth 1 point, right? So to try to avoid that, just get all the easy questions out of the way first and then focus on these other guys. Okay. The results of the experiment suggest that, okay, let's look at B. The change in, so the mutant and wild type differ in their CT1 binding site. Uh, the wait, what is this even saying? Delta this guy and wild type this guy differ at their CTA1 binding site. Mm, that's not correct because these guys are separate from CTA1, right? They're not necessarily connected. I just want to confirm that really quick. I didn't what's it called? Go over. I just went over the correct answer. I'm sorry about that, guys. Let me just run it through real quick. I believe that's the reason why it's wrong, but I just want to double check for you guys. Okay, so is unable to interact with ARFP1 binding site because the structural disruption of the alpha helix at the GTP binding site. Okay, now we're looking for B. The experiment does not test the ability of either wild type to bind to, exactly, right? So these things are not, they're not interfering, right? The RF1 and the wild type differ at their CTA1 binding site. Again, the only thing that it's really looking at is its GTPase activity, right? The GTPase of these wild type things. So for this reason, CTA1 is the classic distractor. It was mentioned here, right, in the passage, but what is the experiment saying? What is the experiment doing? It's looking at the kinetic parameters for GTPase catalyzed activity, right? Not CTA1. What does CTA1 do again? This thing, right? This whole kind of idea here of moving the hydrogen around. That's not, this moving of hydrogens and stuff. Is that GTP hydrolysis? No, it's not, right? GTP hydrolysis is turning GTP into GDP. So look out for that, right? Sure, it says that it's not correct, and that's why I fell for it the first time. I was like, oh look, these two things we're talking, like these two things were mentioned. I'll choose it, and I left, and I moved on. But that's not correct because what is this thing all about? GTPase activity. This thing is it GTPase activity? No, it's not. Okay, so continue forward. All right, here in C, ARF1 gap decreases in GTP binding affinity of ARF1. Okay, so let's check it out. It says it decreases the binding affinity. Which one of these two are binding affinity? This one, right? So a decrease in binding affinity would mean that you have a higher Km, right? Remember, a low Km is good, a high Km is bad. So this thing, right, this assistant guy, it says that it decreases the affinity of this of ARFF1. Okay, so that's cool. Like we have a wild type version and where this thing is present, and then a wild type version when this thing is absent. So let's compare these two. Okay, when it's present, we have a 1.4 km, pretty low, right? When it's absent, this thing has a high km. That's bad. That sucks, right? It has a higher km. If it has a higher km, that means less affinity. So this thing is wrong, right? AF1 decreases the GTP binding affinity. No, it actually makes it really good. It has a low km. Okay, so you can cross that one out. Okay, D. The maximal rate of GTP hydrolysis by this guy is less than that of the wild type. Okay, so let's check it out. It says the wild type is here. The maximal rate of GTP hydrolysis by this guy is less than that of the wild type. Let's just look at the numbers. Let's look at this number here. Is this less than this number? Is it less than this number? No, this number is the biggest one. So this would be wrong as well, right? So then it says when it says maximal rate, this thing is not telling you rate, right? This is just telling you affinity. This thing, the catalysis, this thing is telling you rate. So we just got to compare those numbers. Is this thing less than? Oh, it's not. It's the bigger one, so we can cross that one out, leaving A, right? Okay, let's look at A. The ability of ARFGP1, so this assistant guy, to interact with the wild type, or sorry, not the wild type, the mutant, depends on an alpha helical structure. Well, where can we derive this information, right? Okay, well, check it out. It says here in the passage that the what's it called? That the mutation occurs where? The mutation occurs in the alpha helix, right? This is right here in the passage. An experiment was performed to consider the effect of a mutation in the gene encoding RF1 at the alpha helical structure of the GTP binding pocket. Okay, well, why is that relevant? Well, let's look what happens when you have a mutation, right? When you have a mutation occur, what happens? The affinity for it decreases, right? This is a bigger number than this number, right? So damn, this number increased. So when it says here, the influence, we can see obviously that a mutation has changed the KM, even in this condition when this thing is present, right? And these two things, this guy is present, but the only thing that's difference between these two conditions, right, this experiment and this experiment, is the fact that there's a mutation, and the mutation causes a difference in KM. So the mutation is relevant, right? The mutation actually does something. Where's the mutation? The mutation is at that alpha helix, which means that this thing is correct, right? The ability of AR1GP to interact with the mutation depends on an alpha helical structure. It depends on it. Why? Because if there's a mutation in it, then what happens? We have a higher KM, which means it's bad, right? It has a lower affinity. Okay, so that's this question. All right, we'll keep on moving. All right, so we have question 45 now. All right, let's keep it going. 45. All right, this is a, what's it called? A freestanding question. Okay, it looks like it's gonna be easy peasy. Which of the following compounds would be most soluble in ethanol? Well, what's ethanol? It's this structure right here, it's just 2 carbons and an OH group. This is a polar group, right? Because if you have a small amount of carbons here, I go into detail like what, how to determine something's polar or not. If you have a small carbon chain in a polar group, that thing's going to be polar. If you have a huge carbon chain and then just one measly little polar group like OH, well, that OH is not sufficient to make that entire carbon group, that whole big one, polar, right? So it'd actually be non-polar. But if you have a small one like ethanol, this OH group is strong enough to make it polar. Okay, basically it's saying which one would be most soluble. Remember, most soluble in ethanol. Well, remember, like dissolves like. So if you're looking for something to dissolve in ethanol, you're looking for something that is polar. Here, methane. Sure, it's small, but does it have any polar groups on it? No, it's just carbon hydrogen. Okay, so that thing is non-polar. Let's look at stearic acid. Okay, stearic acid, look how many carbons it's got, right? It's got a bunch of carbons. It's got 17 carbons, 18 carbons, and then you have a carboxylic acid group here. This huge thing, even, okay, so if the hydrogen popped off and you got a charge, sure, it'd be a tiny bit, a little bit polar, but this whole thing right here, would it be most soluble? I don't know, right? Because this huge carbon chain kind of negates the fact that there's a carboxylic acid at the end, right? Most likely it's going to be non-polar, even with the loss of the hydrogen. Okay, so let's check it out over here. The 1-chloro-octodecane. Look at that. It's a huge structure, right? And there's only what? 1 little chlorine on it. Is that chlorine going to be sufficient to make that thing polar? Nah. Let's look at formic acid. Let's draw it out. Formic acid looks like this. A single carbon and an acidic group on it, right? This thing is super polar, right? It's even more polar than ethanol. So it's going to dissolve in it. Yes, right? Okay. All right, let's continue forward. We have next question 48. All right, let's check it out. All right. I just want to make sure that everything is recording, because if it wasn't recording, I'll probably just die. All right, it is recording. Awesome. Okay, let's check it out. We have ChemPhys, question 48. Perfect. All right, let's look at it. It says here, which indicator would be most useful in signaling the second equivalence point of a typical neutral amino acid in titration used with NaOH? Okay, basically, it's looking for a indicator. What does an indicator do? A good indicator changes color when you reach a equivalence point in titration, right? So you reach your equivalence point, let's say the indicator goes from red to blue, right? That's awesome. If the indicator turns red to blue at the equivalence point, then we'll be able to know when we reach the equivalence point. You really need to have a good understanding of what titration is to appreciate this question, right? So, okay, let's go over the fundamentals really quickly of what a titration does. Basically, what a titration does is kind of try to see how concentrated something is, or how acidic something is, or how basic something is, right? For example, if you're just looking at for concentration, then what you would do is that you'd have some sort of reactant in a mixture, and then you'll add something to it. It's something called a titrant. And then when you keep adding it and keep adding it, eventually you'll find the equivalence point, aka the point when the amount of reactant you have and the amount of substrate you have are equal. and then your indicator will just change color. And then, okay, cool. I just put like, let's say X amount of drops into the reactant, and then the indicator kind of changed color. Okay, how many drops did I do? Okay, two, let's say 3 drops. Okay, I know how much concentration is in 3 drops. Perfect. Now I know how much initial reactant we had in that, let's say, in that vial, right? But when we're doing acids and bases, rather than some sort of reactant, we're working with acids and bases. So in our solution, we'll have like, let's say, for example, an acid, let's say an amino acid, right? And the amino acid, like how can we tell how much concentration of amino acid we have? Well, what's a property of an amino acid? The property is the capacity to donate, what's it called? To donate protons. What is that? That's an acid, right? The ability to donate as a proton is an acid. Okay, how can we get the protons off of the amino acid? Well, we can introduce the titrant, aka the in this case the base, right? The NaOH, and NaOH is a base. So we're going to keep adding base until we reach an equivalence point. So when the amount of base that we added equals the amount of unknown concentration of acid, and then a good equivalence point would go from what, one color to a different color. So it's like, okay, perfect. I put 4 drops of the base. That must mean that there's 4 drops worth of acid in our solution, right? But the thing is about, that's good enough for if you had a monoprotic acid, right? A monoprotic acid means that the acid has like just a single proton to give, right? For example, HCl, that's a monoprotic acid. What's a diprotic acid? H2SO4, right? H2SO4, the first proton, very acidic. It'll come off really easy peasy. The second proton, like let's say HSO4 minus, once you take out one of the protons here, proton is a hydrogen, removing this proton is going to take more effort, more base than it would for this proton. This proton is readily easy to leave, but this proton is going to be more attached to this group because of the that's a lot of chemistry to go into why it'll be more stuck onto. I think it just takes more what's it called, kind of. Okay, so let's think about acids, right? So what is an acid? Something that can easily donate its proton. Why would it donate its proton? Because when it donates a proton, it's kind of stable, right? The conjugate base of a strong acid is a weak base. If it's a weak base, it means it's stable. It doesn't want to react with anything. However, if we were to take out another proton, what would happen is that, let's say we remove the proton from here, right? What we get is SO4 2 minus. 2 minus, does that seem stable to you? Like 2 negatives on something? Nah, right? So that's why it's harder for this proton to move than this proton, right? If you remove this proton, you only have 1 single minus. Okay, that sulfur group can, and those oxygens, they can handle that. If you have 2 minus, it's going to take more base. And for that reason, we have like, if you guys remember, what's it called? Titration graphs here at this kind of linear line right there, like this kind of like slope right here, we have an equivalence point of our first proton, right? We have 2 protons that can be donated in a classic amino acid. Remember, the classic amino acid has what in the backbone? It has a carboxylic acid group, so that'd probably be the first proton donated. And then we also have another proton in the amino group, so that'd be the second proton donated, right? So here we have our first equivalence point when all of the protons from the carboxylic acid is removed, right? How do we know that the amino group is not donating protons? How do we know that? Well, because at this pH, at pH of 6, or remember the amino group, its pKa is 9, right? So the environment is more acidic than the amino group. So is the amino group going to donate its proton? Nah, right? Because the environment is too acidic. However, at the pH of 6, the pKa, or the strength of the acid of a carboxylic acid, is like around 2.3. So yeah, this environment is way more basic than the acid being carboxylic acid. So it'll donate its proton. Okay, let's not get lost in the sauce here. This will be the first equivalence point. We keep adding pH. Eventually, we'll get to the second equivalence point when the amino group is going to be donating its proton. Okay, see the first here and the first equivalence point here, and we have this kind of like, you see how this structure right here is similar to this structure? That's because our second equivalence point is going to be up here, just not drawn. Right? Okay, so if our second equivalence point is all the way up here, remember, we want our indicator to change at the equivalence point, right? So that means that we're going to have an indicator with a pH range that's higher than 10, right? Because the slope over here, if we were to keep drawing this, right, like over here we have it drawn over here. Like this is the first equivalence point. The second equivalence point will be like over here, right? And then it'll keep doing that. So we're going to want something that has a pH that's, or like more than like a kind of like towards 10, like a higher pH. And which one is that one? It's this yellow one, Okay, perfect. This yellow one. I think I guessed on this question. I was like, fuck it. But yeah, this would be the correct one because it has the pH range higher. And remember, we want the equivalence point to change at the equivalence point. So if the equivalence point is like, let's say 11, then the best one to use is what? This one, right? Because it will change at 11, right? These guys won't catch it. These guys would change way beforehand, and that's not going to be accurate. This guy being more accurate would be the best option. Okay, let's continue forward. So our next question is question 53. Yep. All right, 53 is over here. Okay, here it says the point mutation denoted by beta represents the substitution of an amino acid integral to the transition state of which of the following pathways. Okay, there's a bunch of stuff here. Let's break it down, right? And it seems like we're going to have to use the passage to figure things out. Okay, so let's see. It says the point mutation denoted. Okay, so point mutation, what is a point mutation? It means that something has changed, right? So a point mutation, perhaps our amino acid is no longer the same as it once was. Maybe it was glutamic acid, now it's valine, right? That's point mutation. So the point mutation denoted as beta, okay, so the point mutation has a name, beta. Why? Well, maybe because there's other point mutations, and this one just happens to be called beta, represents the substitution of an amino acid. Okay, yeah, makes sense, right? The point mutation. Integral. So, okay, whenever you see things like integral or super important or most important, whatever, you're looking for an extreme, right? Typically, whenever it's asking you, like, okay, in which scenario would this thing happen? Typically, you're looking for an extreme. And I know before we mentioned that, like, oh, an extreme was bad. But remember, that was just in the case when I believe it was it this one? No, yeah, 25. In this case, when we had the extreme 25, the reason why it was extreme was because it went outside of the scope of the information provided by the graph, right? I believe it was this one, lipid hydrolysis cannot occur at all. So yeah, exactly, right? This thing, the lipid hydrolysis cannot occur in the absence of raclopride. Well, we only know what happens when raclopride is there and when it's not there. We don't know if a different acid can do something else to it, right? So that's extreme in terms of it's not like the information there doesn't give us the capacity to make that statement. However, in question 53, it's asking us, which is the thing that's integral? So whenever something is integral, let's say like we had a number line, right? Like number 10 is the most important and number 1 is the least important. Are we going to choose something that's over here? We're going to choose something over here? No, we're going to choose the thing that's most extreme. Why? Because it's integral. Whenever it says integral, okay, we're going to look for the most important thing. Like, why bother with these other guys that are not the most integral, right? This thing right here, in terms of importance, 10 is the most important. So we're going to be looking for the thing that is an extreme in terms of like how important it is, right? We're not going to choose something that's half-assed. In the transition state, oh, a transition state, I know what that is. Kind of sounds like an enzyme, right? Like remember in enzymes, we had a transition state like here, like this would be the transition state. This would be change in G, right? The reactants have this much free energy, and then sorry, not change in G. Like this would be G, like free energy. The reactants have this much energy. The products have this much energy, right? So if the energy decreased, that means that we went from something that was unstable to stable. If we had something different, like if we had this, for example, oh shit, this is kind of weird. We had this shit. If we had this, and this is our transition state right here, then guess what? We're going to run for something that had a low Gibbs free energy to something that had a higher Gibbs free energy. Well, that's not good, right? That's from low energy to high energy. Remember, we don't want to be in high energy as a molecule. So this is something that would be like non-spontaneous. This thing would be spontaneous because we want to go from low to high. Sorry, we go from high to low. This one non-spontaneous because we go from low to high. Not good. If we're going to high to low, it is good. Okay. That's something that I explained a lot before, so just definitely in the PowerPoints, go definitely check it out. But the thing I want to get at is like right here, this kind of like energy, because remember this is energy. So this thing right here, this energy gap right here, that's your transition state, right? Like sorry, that's not your transition state. That's actually your activation energy, how much energy you need for the reaction to occur. This peak right here, that's your transition state. That's the high level, high energy intermediate that lives for a little bit, and then it goes from that high energy intermediate to your product, right? Okay. So saying here, the transition state for which the following pathways. Okay, so we have to look for beta, right? Where beta is important. Okay, let's scan our kind of place for where beta is introduced. Okay, so it's blah blah blah. Anything about beta here? Okay, nothing. All right. Anything about, so we're looking for the basically any language that deals with beta, transition state, or P, right? That's the thing that we're looking for. Any of that shit here? Nah, right? Okay, so how about over here? Blah, 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 blah. Oh, we find P. Specifically, those P's that we're talking about, right? P1, P2, P3. And here we found the P's. Okay, cool. So let's read this paragraph. A specialized molecular tool known as optical tweezers allows the participant to, okay, blah, 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 sounds like biology. Who cares? Okay. Different kind of mechanical stress, elongation and shearing. Okay, so there's mechanical stress. There's two different kinds, elongation and shearing. Okay, cool. I'm going to take that into note. On a Pico-Newton scale, really small force, to single protein molecules. Okay, so basically we can apply using optical tweezers small amounts of forces as either elongation or shearing. Okay, cool. That's all I need to know. Application of these forces the application of these forces induced the protein to unfold along different pathways. Okay, so if you can imagine that, like let me show my camera. If you can imagine that, like let's say we have a protein and it's like this, right? If we do elongation, it kind of shows us what a graph of elongation looks like right here. Right, this is elongation. It's like pulling in this way, pulling in this direction. So you get elongating it, right? You're going for something that's like have a firm kind of structure, right? This structure gives the protein its function, right? So if you change the structure, so let's say we had this, but if we were to elongate it like that, well then the function is changes. It's become sort of denatured, right? That's elongation. And here for shearing, instead of pulling it diagonally, it's kind of like pulling it kind of up and down, right? So that's the two different kinds of forces that we're dealing with here, right? Let me turn my camera off. Okay, so all right, so here we have elongation, shearing. Okay, application of these forces induce the protein to unfold along different pathways. Okay, so it takes different pathways. The elongation factor, okay, one of those two kind of forces. The elongation forces are applied. Figure 1A. Okay, so figure 1 here. Figure 1A would be this one. The elongation forces are applied, figure 1A, only, okay, when elongation forces are applied, comma, there should be a comma here, only one transition state geometry was detected. Okay, so transition state, which means we're going to have, yep, and we do. We have free energy as our y-axis, and here we have, when it says extension coordinate, it just means time, right? So, all right, so here we have, when we have zero force, okay, there's nothing be applied. When we have zero force, guess what? Our transition state, our transition is pretty big, right? We go from over here to all the way up here, and eventually we're ending up over here, right? So that's that transition state. It's really just the difference between, like the net difference. Like, okay, so here we have the one over here, and then that'll be the difference between the reactants and products. Anything else above that, like this stuff right here, that's your activation energy, aka your transition state is over here. When you have force applied, though, look what happens. We go from something that was actually non-spontaneous. Check it out. Like this thing was non-spontaneous, right? If you have something that's low to high, remember low to high is non-spontaneous in terms of energy. You want things want to be low energy. So if you're making it high energy, it's this reaction it's not going to occur spontaneously, right? Something has to happen to it for it to actually occur. And what's occurring here? The reactant is what? The folded protein. What's the product? The kind of denatured unfolded protein, right? So for this thing to become denatured on its own, remember zero force applied, that thing is non-spontaneous. It's not going to happen on its own. However, if we have force being applied, right? What happens? It goes from something that is high to low. So that's actually something spontaneous, right? And guess what? The transition state decreased. Okay, so it's like you don't even need that much energy before, right? For this one, you need a bunch of energy for this thing to happen. This one, not that much energy, right? When we have a force of 10, that's elongation. Okay, cool. Let's continue forward. All right, this indicates that only one pathway is present, right? P1. Oh, okay, so that's P1. P1 is when we have elongation. Okay, cool. All right. Okay, this indicates that only one pathway is present. The same pathway followed when unfolding is only induced by chemical denaturing. Okay, so it's either elongation or chemical denaturing that causes this P1, this pathway 1. Okay, under shearing forces, okay, remember elongation and shearing. Shearing is one of those two forces that can be caused by the optical tweezers. Under shearing forces, however, of varying degrees, two new pathways were detected. Okay, so we're not going to follow this one, right? This one was the initial pathway 1, well, for elongation. But if we have shearing forces instead, we're going to have different pathways. Okay, so let's check it out. We have two different pathways, P1, P2, and P3. The geometries of the respective transition states are denoted by TS1, TS2, and TS3, right? Here's TS1, transition state 1 for pathway 1. Here we have TS1, again, just reiterated. And then here we have transition state 2 and transition state 3 for P2 and P3. Okay, all right, so cool. Let's just interpret this graph real quick, right? Okay, so here we have 0 forces, and here we have pathway 2 in the dots, and pathway 3 is dashed, and P1 is in the straight line. Okay, so here we have P2, not really high the transition state, so okay, so that's pretty good. You know, they all end up what? Denatured, right? They all end up denatured. So which one seems to be the most kind of improbable thing to happen? Kind of P3, right? Because P3 needs so much energy for it to happen, right? So much energy. P2 less energy, P1 even less energy, right? When you have 0 force applied. When you have some force applied, let's look at it. We go from folded to unfolded, denatured. What happens when you have 7 force? Well, it seems like things are kind of more equally distributed, right? The P2 is kind of close to the P1, and the P3 still, the P3 is super OD what? Like not good, right? It needs a lot of energy for it to happen. But let's look what happens when you have force of 25 piconewtons, right? So that's a lot of force, right? So it's more force than 0, it's more force than 7. See what happens. 1, that thing needs a bunch of energy. 2, pathway 2, remember pathway 2 is the dots. It needs some energy, but look at pathway 3. Pathway 3 used to be the problem, right? It used to be like, oh man, it's not going to happen with 0. Like it's really hard for it to happen here. It's really hard for it to happen here. But here, the transition state, and now it's super duper low, right? It's like, whoa, it's actually, you know, we need if you want to pathway 3 to occur, it'd probably be best occurring when you have a bunch of force attached to it, right? The transition state is really low. Okay, so now that we have that figured out, where does beta come in? Like, where does beta come into play? All right, so let's keep reading so we can find beta. Okay. To gain insight into the areas of protein structure involved in each transition state, okay, so we just want to like know what's the kind of, remember, the structure gives you function, and so we're just trying to look at the protein structure, right, for each transition state. Like the transition state is this peak right here. They're just trying to look at what the protein looks like in this high energy state. Selected point mutation, okay, point mutations were changing amino acids, were introduced into the peptide chain, and the changes induced in the EA just means the activation energy. Remember the thing that over here, like this is your activation energy. This is the energy needed in order for the reaction to occur. If you can't reach activation energy, the reaction is not going to occur, right? What does an enzyme do? Why is an enzyme so nice and useful? Is because the enzyme, it kind of decreases the amount of action, activation energy that you need, so it makes things happen more faster, right? Let's say you needed 100 volts of energy or 100 joules of energy for a reaction to occur. Well, the enzyme just means like, okay, now instead of needing 100 joules for the enzyme for the reaction to occur, now you only need 10 joules for the reaction to occur. So that's why enzymes speed things up, right? It just makes the activation energy decrease. Okay, induced in the activation energy of unfolding were measured. In figure 1c, each Greek letter alpha, beta, gamma, delta, epsilon represents a specific point mutation made in the peptide, and the value, I think that's psi or phi, psi, represents the effect each mutation had on the transition state under the given amount of shearing stress. Okay, so let's look at our figure 1c, right? Here we have this thing psi. How much, what's it called? Psi was the value. Psi was the represented as the value that has the effect. Psi represents the effect each mutation has on the transition state. And here we get a better definition of psi. The psi value ranges from 0 to 1. Values close to 1 indicates a large effect on the observed transition state. 0 means that there's little to no effect. Okay, so remember, what is the question? Ask for the integral thing, right? So we're looking for something that really alters things, right? Remember, it says here's that value, a psi of 1 means that what? It indicates a large effect. So we're looking for something that's integral. We're looking for something that has a large effect. So we're looking for something that's close to 1, right? Okay, so, and then we have alpha, beta, gamma, delta, epsilon. Which one are we referring to in the question set? Beta. Okay, so we're looking for when beta is at its highest, right? You're looking for beta when it's at its peak. Beta is at its peak when we have this beta. This beta is when it's closest to 1. Force is on the y-axis. Okay, so when we have a bunch of force and we have the beta mutation, we have a huge change in what's going on in the transition state. Okay, so now that we know this information, now that we know that beta is really important whenever we have 30 piconewtons of force, well, which pathway, P1, P2, P3, had the kind of transition state be really kind of like changed when we had a bunch of force? Right here, the force is 30. Well, if we go back to this, the transition state here is kind of high when you have 0 newtons. or sorry, 0 piconewtons, and then when the piconewtons is 7, we have the transition states are kind of the same. But we have a huge alteration in which pathway? In pathway 3, remember, when we have 25. So imagine if we had 30 newtons, sorry, 30 piconewtons of force at the beta. Well, guess what? We're going to have a huge change, right? Because it says here at 30 here, when you have a little bit of force, beta doesn't really play a part. But when you have a bunch of force, beta plays a huge part. And so for that reason, P3 would be the correct answer. Okay, remember, if you have any questions, go through the slides. I have it written down here in this paragraph. But yeah, definitely reach out if you guys need help with that. Okay, so next question, question 54. All right, let's check it out. 54. I didn't even answer this question. Probably ran out of time or something. Okay, which of the following is the best explanation for why urea may be used to study the unfolding of blah? Okay, so we're gonna have to find blah, right? Okay, where's blah? Let's look for it. Oh, here it is. All right, so it says here. Let's see if we need the stuff from the 4. Okay, let's just read it. Okay, in the vast majority of studies of the unfolding and folding of proteins, the unfolding has been observed to follow a single pathway characterized by one transition state. Okay, we kind of already knew that. However, this alone does not rule out the existence of multiple unfolding pathways. Okay, like P1, P2, P3. As it is conceivable that only one pathway is kinetically favored, okay, the transition states, whatever, under all of the experimental conditions employed, making it is the only one that's observed. Okay, that's really convoluted. Do we need to know that? Not really. In one study, the unfolding of a well-characterized protein domain, okay, this guy right here, was part of the question stem, was examined in the presence of combination of chemical denaturants. Okay, what's a chemical denaturant? It's a chemical that denatures proteins. What is something that denatures proteins? Something that you should have memorized is urea. Urea is something that denatures proteins. How does urea denature proteins? Well, urea denatures proteins by interrupting the intramolecular forces and interacting with polar and charged regions, effectively preventing them from interacting with other amino acid residues as it should be. What does that mean? Basically, our amino acids look like this, for example. They have hydrogen bonding. They have other things interacting between, like, let's say you have a beta plated sheet, right? And this is one chain of amino acids. This is another chain of amino acids. We have a sheet, right? Whenever we have folding, our primary structure is a chain of amino acids. Our secondary structure is an alpha helix or a beta sheet. Let's say we have a beta sheet. Well, what urea does, urea is this. Nitrogen. Okay, this is an ugly looking urea. So this is what urea looks like. Nitrogen on here, nitrogen over here. These nitrogens are what? They have the capacity to hydrogen bond, right? So basically urea comes over here, kind of goes through the middle of this guy and fucks things up, right? It's a chemical denaturant and messes up proteins. That's something that you guys should know. The rate of unfolding must alter with changes in the concentration of chemical denaturant, aka urea, according to this equation, blah, blah, blah. Okay, seems like we have everything that we need to know. It is here, which of the following is the best explanation for why urea may be used to study the unfolding of this guy? Well, basically, remember, if we have unfoldings, it's if we don't have any force involved, well, guess what? It's going to be something that is unspontaneous, right? If you have no force, it's going to be something that goes from low energy to high energy. That's non-spontaneous. So if you want to make sure to kind of keep that unfolding, that denatured state, we're going to have to do something, right, to keep it in that state. Well, what can we do to keep it in that state? We can introduce chemical denaturants like urea to keep it there so we can study it, right? Let's look at the correct answer. Urea acts to stabilize the denatured state of blah, blah, blah by associating with the backbone polar groups and charged side chains. Perfect, right? Basically what we just talked about, that would be the correct answer. Everything else pretty much kind of gets the idea of urea wrong, right? The addition of the urea reduces SS. Urea is not going to reduce jack. Urea stabilizes the multiple transition states observed by for the unfolding of this guy. It's not that it stabilizes the multiple transition states observed. Okay, what I see is wrong. Okay, let's see what I see is wrong. As stated in the passage, only a single unfolding pathway is followed okay, right. So like a bit of information that we didn't catch. But again, if you know that urea is a denaturant, as soon as you read B, guess what? You're choosing that thing, right? As soon as you know that urea is a denaturant and you know the mechanism, why is it denaturant? Because it interacts with polar groups and side chains, charged side chains, then seeing something like this, like it's gonna be like, okay, well, B sounds better to me, right? And I don't have enough time, so I'm gonna move on, right? You got to keep it real, right? You're not gonna know everything on the spot. You're gonna have to use the process of elimination. This thing, if you didn't read, like I didn't read this thing carefully, right? I just read to get the point. If I missed that it was something about multiple transition states, but the answer choice is like, no, it's actually only one, not multiple transition states. Well, guess what? This thing is a better one. I'm not sure about this guy. Time's running out. I'm choosing this one, right? And it's the correct answer. And then okay, that's what you do during the exam. How can you prevent that from happening again? You do some really nice error analysis at the end, right? You do error analysis, you figure out why this and that, your mentality, etc. Okay. And then the entropy loss due to unfolding of blah blah is a result of urea increasing the solubility of the protein interior. It's not that it's increasing solubility; it's just breaking them apart. Let's see what the TPR has to say about that. Choice A presents an inconsistency because unfolding will cause entropy to increase, not decrease. That's true, due to the disordering effect of unfolding. Yep. Entropy is entropy is just disorganization. I didn't read that carefully. The entropy loss due to so entropy, like it's saying that disorganization was lost due to unfolding. Well, if you have something that's folded and turned into something that's unfolded, right? You had something that's like a ball and then you turned it into like a sheet of paper. Well, guess what? Like that thing that was one nice and compact, but now it's more deorganized, right? So you actually have a gain of entropy rather than a loss of entropy. So yeah, right. You just want to. That's more chemistry than biochemistry or orgo, but whatever. And then urea right here, right? That'd be the correct one. Just know that urea denatures things. How polar groups side chains answers the question. Why would it be useful here? You're good to go. Okay. I think moving forward, I'm gonna have to like read the other answer choices just so that I'm prepared, because some of those answer choices can be tricky. All right, for ChemPhys, question 26, the reason why we backpedaled from 53 is because this is something that I categorized as not peasy. Remember, we had things that were less peasy or easy peasy. See question 26 and question 34 were things that were not peasy, right? Okay, so let's look at question 26. It says here, which lipid would have the smallest y-intercept on the Lineweaver-Burke plot of the kinetics for raclopride, blah, blah, blah. Okay, so it's saying here which lipid. So there's multiple lipids. We're not going to be looking at DOPC because DOPC is only one lipid. We're going to be looking at all of these guys. And let's look at the graph. Okay, so the graph has rates as the y-axis. Typically, you have something like concentration here as your y-axis, but here we have rate. This is rate of lipid hydrolysis versus saturated lipid concentrations. All right, so here we have rate on the y-axis. Here we have dopamine and lipid concentration over here. So basically, for most of them, the higher the concentration, the faster the rate, right? Okay, so it's saying which lipid will have the smallest y-intercept on the Lineweaver-Burke plot of the kinetics for raclopide-mediated lipid hydrolysis. Well, all of this stuff, everything here, like look at the legends, all this stuff was raclopide mediated, so we don't have to worry about choosing one. Like they're all valid. And if you guys know Lineweaver-Burke plot, something in that deals with enzymes, you'll know that the smaller your y-intercept. So whenever they're talking about y-intercept or x-intercept, they're talking about the Lineweaver-Burke plot. This is the Michael-Mentis graph. Like this, the information here can be altered to make this graph, and this graph is just more kind of like it gives you a little bit more information without having to like strain your eyesight or whatever. Basically, in this graph here, to interpret it real quick, the x-axis is concentration and the y-axis is the rate of the reaction. When you have the highest rate, you have Vmax, and yeah, that's all you really need to know. Well, guess what? If you have a high Vmax, right, and if you look over here, the y-axis in turn is no longer just Vmax; it's 1 over Vmax, or sorry, 1 over velocity. Well, guess what? If you have a really big number, like let's say 100, as your denominator, you're going to get a really small number, right? But if you have a small number as your denominator, you're going to get a slightly bigger number. All right, sorry, 0.1, right? So basically, when you have when you're looking at y-intercept, the smaller the y-intercept, the actual bigger the Vmax, right? Because remember, we just discussed this. You have a really big number as your denominator and you divide it by something, it's going to be a small number. If you divided, like, instead of 1 over 100, like I say 1 over 1,000, right, it would be 0.001, right? So a smaller number, right? So basically, the bigger your Vmax, or the bigger your rate, V just means velocity. The bigger your Vmax, the bigger your rate, the bigger the number, the denominator would be in the Lineweaver-Burke plot. Because look at the y-axis, 1 over V naught, right? So V naught just means velocity. So when the question is asking us, hey, which lipid will have the smallest y-intercept? It's basically asking us which one has the what? The biggest Vmax, right? Because the smallest y-intercept is correlated with the biggest Vmax because the equation used is 1 over Vmax. If you have a big number on the bottom, you're going to get a small number as your answer, right? When you do your division. Okay, so it's saying which one has the biggest rate? Can we look at these? The y-axis tells us rate. Which one has the biggest rate or the fastest rate? Guess what? It's this one, and this is DPPC. So the correct answer would be DPPC. And I just put, and I was, whatever. Yeah, okay. So yeah, DPPC would be the correct one. Okay. Now let's go into the next one. All right. Our last one for Chem Phys would be question 34. Let's check her out. Question 34. All right. This guy again with the NADPH and the glutamine groups that kind of snatch hydrogen. Let's see what's going on here. It says here, the change in the kinetic parameters of ARF1 following treatment with the experimental ARF1 inhibitor. Okay, so we have an inhibitor. We have ARF1, which is an enzyme. ARF1 inhibitor is something that inhibits RF1, is the same as the change measured after replacing the wild type gene encoding RF1 with the mutant gene. Okay, basically, what is that trying to say? It's saying that the effects, trying to say is that the effects of the ARF1 mutant, so that whatever the effects that this is, has the same effects as the ARF1 inhibitor. Which means that we're going to be looking at what? This graph, this data here again, right? So let's look at the wild type and let's compare that to the mutation, right? Because that's basically the wild type without inhibitor versus the wild type with the inhibitor, right? Because the inhibitor and the mutant have the same kind of results. That's what's saying over here. If we look at the wild type, we have what? Km and we have the Kcat. When we have the mutant, look at the Kcat, look at the Km. The Km has increased, but the Kcat hasn't really appreciably kind of increased. It just increased by 0.01. And not only that, but look at these error bars. The error bars include this number, right? If you add 0.017 to 0.23, we're going to get 0.24, right? So that's what these error bars mean. If these error bars overlap with one another, that means that they're really not significant, which means that these two numbers are really not significant. So Kcat really doesn't change with the mutation, aka doesn't change with the inhibitor, right? Because the mutation and the inhibitor are the same thing. What changes is the Km. Okay, so there's different kinds of inhibitors. You have the non-competitive, you have the competitive, you have the mixed inhibitor, and you have the uncompetitive. These are things that you should know from the enzymes. When you guys were outlining your biochemistry book, you should have touched upon this. I'm also going to be releasing a video on this pretty soon, right? I've been really busy with the Audaces. But basically, okay, so which inhibitor only changes the Km in a negative way, right? So an increase in Km, that's bad because the lower Km, the more affinity you have, the higher the Km, the less affinity you have. Okay, so what's something that kind of messes up the Km for your enzyme? A competitive inhibitor, right? So the competitive inhibitor, think about it. The competitive inhibitor, what it does is that it goes to the active site of the enzyme, and what a competitive inhibitor does is that it goes into the active site and kind of occupies that space. So if you have that enzyme and it's occupying space, well, guess what? The substrate won't have an easy access to that space, right? So the substrate can replace the inhibitor. I can shove it off, but it takes a lot of effort, right? So basically, the competitive inhibitor, it decreases the affinity the substrate has to the enzyme. Why? Because the inhibitor is in the way. It's in the active site. It's blocking it, right? So the substrate, it can kind of like maneuver its way in there and kind of knock it off, right? If there's a high concentration of substrate and a low concentration of the inhibitor, well, guess what? The substrate is going to outcompete the inhibitor, right? But at the same time, the inhibitor is kind of decreasing that affinity, which means if you have a decrease in affinity, you're gonna have an increase of a Km. If Km is the only thing that changes, Kcat doesn't change, well, guess what? You have a competitive inhibitor. Because what does Kcat mean? Kcat means that it's the enzyme's ability to create product. The competitive inhibitor being there is not going to affect its ability when the substrate binds, right? When the substrate binds, that's when the Kcat is there. The Kcat is going to turn into product. So when the Kcat, so when the substrate is on the enzyme, it doesn't matter the competitive inhibitor is somewhere out in the environment. It's still going to cause that reaction. So the Kcat is not going to be altered by a competitive inhibitor. Other types of inhibitor are going to affect the KCAT and the KM differently. We'll discuss that in the video, or you should just know that from your outlines. All right. So that's it for ChemPhys. The next one will be BioChem. Okay, let's check it out. BioChem, we have question 59. Okay. So here we have the oh guys, I'm starting to slow down because I haven't eaten today. I'm just starving. I woke up like at 10, and then like I did some stuff for thesis. Kind of like pooped out, but let's keep going. How many questions do we have for BioChem? Oh snap! We got 1, 2, 3, 4, 5, 6, and 7. I'm gonna stop this video here. I'm gonna grab some lunch, and then I'll have like multiple videos up. But you know what? That's not convenient for you guys, so I won't. Okay, push on through. Okay, so it says here, if the measurement taken in step 4 of experiment 1 and 2 had shown no change in the bacterial lactose concentration, then it is most likely what happened, right? Okay, so it seems like we're gonna have to use the passage to answer this. Okay, let's go to the passage. Here we have, okay, so it talks about the no change in bacterial lactose. Okay, so it's scan for lactose, right? Okay. Blah, 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 scanning for lactose. Oh, it seems like it's even here in the first paragraph, in the second paragraph. Actually, here, is it in the first paragraph? It's not. Okay, so it's in the second paragraph. Okay, cool. So you need to forget about the first paragraph. Okay, so this is here. It is unknown that the single-celled E. coli, it is known that the single-celled E. coli organism has the capacity to concentrate lactose intracellular. Okay, so it has the capacity to have lactose inside, right? Remember concentration gradients. If you have, just to reiterate a concentration gradient for like the 10th time, if you have a bunch of stuff here and you only have a little bit here, if you have a membrane separating the two, then concentration gradient, basically what it does is through osmosis, it kind of pushes the high concentration to the low concentration just to even things out, right? So let's say 5 each, right? It prefers to be like that because there's a more stability like that, right? So kind of like the same thing with our proton gradient during ETC, right? We have a bunch of protons here, and this proton gradient kind of causes the enzyme to rotate once it comes down its concentration gradient from a high concentration proton to a low concentration proton, right? So that's just concentration gradients. It's saying here that it is known that a single-celled E. coli organism has the capacity to concentrate lactose intracellularly against its concentration gradient, right? That's something that's really cool because what it's saying there is that even though there's a concentration gradient present, that the osmosis is not going to occur because the E. coli is going to hold on to its lactose, right? A group of investigators wish to study the mechanism through which this concentration of lactose is achieved. Okay, let's check it out. And now we have the experiment, right? We didn't need to read the first paragraph. Cool, we dodged that. Okay, the experiment 1. Okay, so step 1, the population of E. coli was incubated with a medium rich in lactose. Okay, so it seems like probably what they're going to go for is they're going to want the medium rich in lactose. They want that lactose to eventually end up in the E. coli, right? They're going to observe how that happens. And intracellular levels of lactose were measured. Okay, so they're measuring, like, let's say the baseline of it is like, let's say 10, and the environment, let's say there's like 100 lactose. The bacteria were then transferred into a lactose poor medium, having a potassium ion concentration that was substantially greater than the intracellular concentration of this ion. Okay, so now as this is the 10 lactose, we put L, 100 lactose in the environment. Here we have, let's say 100 potassium, and here let's say we only have 1 potassium, right? That's the thing that they described here, right? The bacteria would then transfer to a lactose poor medium, so there's no lactose involved in the medium, having potassium concentration that was substantially greater than the intracellular concentration of this ion. So basically the potassium in the environment is huge, and the potassium in the cell, like this is the cell right here, right? This inside, this is the cell, this is the environment. Well, guess what? The potassium in the environment is greater than the one inside the cell. So we're going to guess what? That osmosis is going to occur, right? Because they told us that osmosis doesn't occur for lactose, but it didn't say that it wouldn't occur for potassium. So we're going to assume that regular osmosis is going to occur for this potassium, meaning that the high concentration of potassium is going to go into the cell from the environment. Okay. And then in this last one, we have as a control, some bacteria were transferred to a lactose poor medium without potassium gradient. So basically, this is just a cell within a medium, and there's no lactose in the medium, there's no potassium in the medium, it's just doing its thing. Okay, step 2. After a specified period of time has elapsed, the intracellular concentrations of lactose were measured and the average values were derived. This value was then compared to the average concentration of lactose within the bacteria at the start of the experiment. Okay, so basically, like this is their baseline, and by the time that the experiment ended, they had different kind of things happen to it, right? There's different concentration involved. Okay, step 3, the concentration of potassium in the lactose poor medium. Okay, so the lactose poor medium was what? Uh, over here, like this one was lactose poor. This one was also lactose poor, but this one's K plus rich, and this one's K plus poor, right? K plus poor. This is K plus rich. This is lactose poor, K plus poor. Okay. So they're talking about this one, right? They're talking about this one in the third step. The concentration of potassium in the lactose poor medium was measured and compared to the potassium concentration in the medium at the start of the experiment. Okay, so basically they're trying to see how much of this potassium went into the cell, right? Because regular osmosis occurs here. Why? Because they didn't say that osmosis doesn't apply, right? Here it says in the second paragraph that the E. coli, the cell that we're looking at, is able to prevent osmosis from occurring for lactose. Okay, let's look at step 4. Steps 1 and 2 were repeated, but instead of imposing a potassium gradient, the investigators imposed a proton gradient. Oh, okay, so instead of potassium K plus, we have protons H plus. Okay, cool. The medium was acidic relative to the inside of the cell, blah, blah, blah. Okay, cool. And then we have the results for our experiment, right? So, okay, so look at the legend here, the effect of ion gradients on E. coli lactose uptake. Let's look at our key here. This is the key is positive means that the concentration increased. Oh, okay, so from baseline, when we go to, after some time passed, what happened was that if we have a positive sign, that means that the concentration of whatever we're looking at increased. If you have a negative, that means that from the baseline, after time passed, the concentration actually decreased. Okay, and if it's 0, it means that there's no change, right? Okay, so let's look at the graph or the table. Here we have an imposed gradient. Remember in steps 1, 2, and 3, we had the potassium, and then step 4, we had the proton, and then we have a control, right? When there's neither, like in this one, there's nothing happening here, right? There's no ion involved. So that'd be like this third one right here, when there's none, like no imposed gradient. Okay, so now they're looking at bacterial lactose. What does it mean to be bacterial lactose? That means lactose within the bacteria, right? There's not the lactose in the environment, just the lactose within the bacteria itself. And then here we have medium ion, right? So what is the medium? The medium is the outside, is the environment. So it's now looking at the concentrations, like whether it increased or decreased. Remember, positive, negative, the medium ion, right? Okay, so let's look at what happens when we have experiment 1, when we have the K plus, like this. This is one line right here in this direction. Okay, so when we have K plus or potassium rich, like this one over here, when you have potassium rich, what happens is that bacterial lactose increased. Oh, that's crazy. Bacterial lactose increased, which means that the lactose poor, so there is lactose present, right? It's just not a lot. The lactose poor, what happened is that with the K plus, in this environment, bacterial lactose actually increased, right? And the medium ion decreased. What does it mean if the medium ion decreased? Right? The medium had, like, let's say 100 potassium, right? If the medium ion decreased, where did those ions go? Those ions went where? Inside of the cell, right? So if the concentration decrease of the medium ion after time, that means that the ion moved from the medium to inside of the cell, right? Decreasing the medium ion concentration. Okay. Let's look at this. Let's look at this correlation here, right? We have the bacterial lactose kind of increasing within the cell, which means that the lactose, whatever amount of lactose is present here, is going to go into the cell. Why? Because the bacterial lactose concentration increased. At the same time that this is happening, the medium ion concentration is decreasing, which means that the ions must be going from the medium to the cell, right? Because the medium ion concentration is decreasing, which means it's shuffling into the cell. So look at that, right? Lactose is going into the cell. The potassium, the ion, is also going into the cell. However, if we have no gradient, which means like we have no K plus or no H plus, when we have no imposed gradient, guess what happens? The medium ion concentration doesn't change. Okay, so there's no movement. And if there's no movement of medium ion, look at this. Bacterial lactose also doesn't change, which means that you need the ion to move into the cell in order for lactose to go into the cell. Because look, when there's no gradient, that means that the osmosis is not going to occur. The ions are not going to be moving. And if the ions are not moving, what we find is that bacterial lactose, there's no change in the concentration. Over here, when we had the imposed gradient, we had an increase of lactose. And even with the H plus, it's an increase of lactose. But when there's no gradient, there's no lactose. So what concept can we pull from our mind that kind of deals with this? Where there's the idea of cellular transport. Is the idea of synport, antiport, and uniport, right? So synport, so there's things in the cell, like let's draw a membrane here. Let's say we had something that easily can just go into the cell without any energy, right? Like it spontaneously can just enter the cell, right? Let's just say K plus. Well, the cell is nice and efficient, and what it does is it's just like, hey man, I have this structure here. The structure here really can't go into the cell by itself like the K plus can. So what a synport does is that it takes the energy, the concentration gradient, the energy from this K plus entering cell, and uses that same energy to move this molecule. So it's just a single kind of, it's a synport, it's a protein. It takes the K plus and it couples it with this thing that can move on its own, and it puts it in the same direction. It's a synport. What is an antiport? Well, basically, antiport is just it's kind of the same concept where it's using the energy of something that can move through the membrane to and pairing up with the other thing, but just moving in opposite direction. So like if we had the K plus here, the K plus is moving in this direction downward, but then the thing that can't move on its own is going to be moving upward, right? It's just an antiport. In this case, we have the ions from the medium going into the cell, and we also have the lactose going into the cell. So we have a synport going on. Okay, seems like we have enough information to tackle this question. All right, check it out. If the measurements taken in step 4 of experiments 1 and 2, so do we need to know experiment 2? Okay, whatever. So experiment 2 is basically it had the, instead of the cell, like the living bacteria, it just had a vesicle, and basically the vesicle acted like the bacteria, right? The same kind of results occurred. Okay. So in this case, okay, so it says these measurements taken in step 4 of experiments 1 and 2 had shown no change in bacterial lactose concentration. Okay, so what happens when we have 0 change in bacterial lactose concentration? That's when we have 0 change, right? Then it is likely that within the medium, what? Hydrogen ion concentration had remained unchanged in experiment 1 and 2. Perfect, right? That's the correct answer. Why? Because when we have no change in bacterial lactose, here, look, bacterial lactose, no change. Bacterial or vesicle lactose, no change. When we have no change in bacterial lactose, as it's saying in the question stem, when there's no change in bacterial lactose concentration, that means that the ion is not also not going to be moving either, right? Because what do you need for lactose to enter? You need the ion to move, right? If you have no concentration gradient, guess what? If you have 50 ions in the medium, and okay, right over here. If you have 50 ions in the medium, let's say K plus, you have 50 ions in the cell, there's not going to be any concentration gradient for the movement to occur, right? So if there's no concentration gradient, the ions are not going to move. And if the ions are not moving, that means the lactose is not moving. So look at this answer. If lactose isn't moving, basically what we can say is that hydrogen ion concentration had remained unchanged, right? Perfect. There's no movement. There's no synport happening. Everything else just gets it wrong, right? The hydrogen ion concentration has increased. No, because there's no movement. Potassium ion concentration had remained unchanged in experiments 1 and 2. The reason, uh, oh my God. Okay. If the measurement of T is taken in steps 4, blah, blah, blah. And step 4, okay, let's read what step 4 has. We're repeating as we said, okay, the medium was acidic relative to the inside of the bacteria cells. Lactose and hydrogen ion concentration measurements were taken and compared to the initial values in step 2. Okay, potassium ion concentration had remained unchanged in experiments 1 and 2. Okay, we have here that the potassium had shown no change in bacterial lactose concentration. It is then likely that within the medium, the hydrogen had remained unchanged. Potassium ion concentration had remained unchanged. Okay, what's the difference between these two? Sorry, guys. I should for next time. Yeah, this is like the smack in the face. This is like a smack in the face. Like I need to, what's it called? Read through all of them just to be prepared. All right, see what's why C is wrong. Potassium ion concentration had increased in experiments 1 and 2. We can cross that one out because if we have no movement of ions, then we have no movement of lactose. Let's look at C. Why is the difference between hydrogen and potassium? It should be pretty much the same thing. It's weird. Okay, so let's see. There might be some nuance that I'm not paying attention to, but okay. The experiments show that the gradient of either potassium or protons can drive the transport of lactose into the cell against its gradient. Yep. If there is no lactose transport, it is safe to assume that the concentration of the media of the driving ion will remain the same. Okay. There is no reason to assume that hydrogen ion concentration would increase, okay, since this is not the case for any of the other experiments. Step 4 involves a proton gradient. Okay, yeah, that's why it's step 4. That's why it's hydrogen ion concentration is correct, because in step 4 we're dealing with hydrogen ions. We're not dealing with potassium. I was kind of freaked out for a second. I was like, wait, what? But it's specifically asking for step 4, which is protons, right? So it's gonna be hydrogen ion. Okay, I was really worried for a second. I was like, damn, where did I fuck up? But it's just these specifically asking for step 4. Yeah, so whenever you have kind of like a situation where you, like I kind of just go like learn from my mistake. Basically, I looked at this and looked at this and I freaked out. Oops, not what you're supposed to do. What are you supposed to do in this situation? You reread the question and you look for something specific, right? Step 4. I read that and I went back to it, but I just kind of like glossed over it. Nah, be specific, right? Steps 4 were repeated, but instead of potassium gradient, we have a proton gradient. And we have a proton here, we have potassium here. That's why this is the correct answer. I fucked up last time, even last year too. I messed up, right? So yeah, if you have two things that seem correct, just go back into the question stem and see where they might be different, right? The difference here is that it's in step 4. Okay, so that's question 59. I don't know why I was drawing on question 48, but okay. So let's just erase that. Okay, so 59, I believe, was this one. That's question 10, 59. Yep. All right. Let's move on to question 10 then. All right, question 10. Says, with reference to the experiment, which of the following did not occur during the 3-minute period following the introduction of catalase? Okay, so let's go to the experiment. Okay, so really quickly, do we need to know what catalase is? Yes. Okay, so catalase are a group of functional and structurally similar iron-containing enzymes, especially prominent in erythrocytes, blah, blah, blah. That's biology. Who cares? They constitute one of the number of groups of enzymes that catalyze the composition of hydrogen peroxide to water. Okay, so that's what catalase does. It takes hydrogen peroxide and it turns it to water. Awesome. Hydrogen peroxide arises from the blah, 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 degradation of cellular waste. Okay, catalase is important to cell survival. Okay, catalase is a high turnover rate enzyme. Okay, it's an enzyme, we know that. A single catalase molecule reacts up to 500 million substrate molecules per minute. That's a high kcat, right? A single enzyme doing all that is crazy. It's a huge kcat. Okay, so let's look at the experiment, right? It's not asking about all this like cell survival shit, right? It's asking us about the experiment. Right, so we just kind of wanted to know what catalase did, right? So okay, let's look at the experiment. In the first trial, hydrogen peroxide, okay, this structure right here is H2O2. What does that look like? And if you draw it out, H2O2 looks like this. So when you have two oxygens bound together like that, that's a hydrogen peroxide, which is just peroxide. In this case, we have a hydrogen peroxide because we have hydrogens attached to it. Okay. In the first trial, the hydrogen peroxide was titrated with potassium permanganate. Remember, if you have a titration, we're trying to add substrate to our solution to figure out how much reactant is within the solution. So basically, this potassium permanganate is going to react with the hydrogen peroxide in order for us to derive how much hydrogen peroxide there is. Okay, so we titrated with potassium permanganate in the absence of catalase. Okay, so there's no catalase. That means that catalase is not converting hydrogen peroxide into water. Okay, in the second trial, the hydrogen peroxide was again titrated with potassium permanganate after the addition of catalase. Okay, so first, in the second experiment, catalase was introduced. And what does catalase do to hydrogen peroxide? It breaks it down. After catalase was introduced, then permanganate was used to kind of, and permanganate, what it's going to do to hydrogen peroxide is just going to convert it into water, right? The following observations were made. Finding 1. 28 milliliters of potassium permanganate was required to titrate 5 milliliters of hydrogen peroxide to water. Wow! So it takes more potassium permanganate than let's say to find the equivalence point. Remember, the equivalence point is when you have the kind of like equal amounts, like in terms of like power, right? If you have something that has kind of a high power, then you're going to need a lot of something that has low power to overcome that, right? So in this case, the potassium permanganate, the power of it to titrate hydrogen peroxide isn't that great. Why? Because it takes 28 milliliters for it to titrate only 5 milliliters of hydrogen peroxide. Okay, let's look at the second finding. 3 minutes after the addition of catalase, okay, so catalase was introduced into the medium, the amount of potassium permanganate required for titration was 1/3 of the amount required in the absence of catalase, which means that, yeah, that makes sense, right? The catalase kind of took away some of the initial hydrogen peroxide. So let's say we had initially 100 hydrogen peroxides. If you were just to add permanganate, then all the permanganate would have to like deal with this, right? In order for a complete titration. However, if we had catalase first, well, what is that catalase going to do? It's going to take some of those things and convert it to water. And then let's say we have 50, or like let's say we had 33 remaining, right? Because it said that let's say we have 33 hydrogen peroxides remaining, right? Because it says here, and the second finding, after catalase was done, it only took 1/3 of the amount of potassium permanganate to titrate the solution, which means that initially it had 100 hydrogen peroxides to deal with, but after the catalase, we only had 33 hydrogen peroxides to deal with, right? So you need less potassium permanganate to actually titrate this thing. Okay, and then third finding, data obtained for the first 15 minutes following the addition of catalase indicated a nearly consistent rate of hydrogen peroxide decomposition, evidenced by the decline in the amount of permanganate required for the titration. Oh, so if it says right here that we have a constant rate of decomposition, what does that mean? That means that the enzymes are doing its thing, and if the enzymes are doing this thing constantly, that means there's no change in the number of enzymes, right? Like if we have 5 enzymes doing work and it's a constant work, if we were to introduce a 6th enzyme, then that constant work, they wouldn't be constant work, it'd be increased work, right? It's saying here in the third finding, however, that during the titration or during the catalase aspect, the rate was consistent, which means that a number of enzymes were the same, right? If you have a nearly consistent rate, that means that the number of enzymes are the same. Okay, let's look at the question now. It says here, with reference to the experiment, which of the following did not occur during the 3-minute period following the introduction? Okay, so basically, it's asking us the second experiment, right? The second experiment is when we had the catalase do its thing. Okay, so let's see here. The substrate bound to the enzyme. Yes, that's what an enzyme does, right? The hydrogen peroxide is going to be bound into the active site of the enzyme catalase. Okay, the substrate enzyme complex was formed, of course, right? Well, actually, hold on. Let me not say of course. The substrate enzyme complex formed. We know this. The reason why we know this is because in the second finding, we find that there's less hydrogen peroxide. What's it called? Available. Oh, which means that that means that some of the hydrogen peroxide were taken out, and in order for that hydrogen peroxide to be taken out, that means that the substrate enzyme complex must have been formed, right? So sometimes a substrate enzyme complex could have been formed, but let's say that after you add the catalase, you still need 28 milliliters of potassium permanganate to titrate it. That means that catalase didn't do jack, right? So the substrate enzyme complex might have formed, but did the reaction proceed? And that's what C is saying, right? The reaction proceeded. Okay, cool. We know that because the number of hydrogen peroxide decreased, right? Okay, lastly, the last one, the one that's correct is a new enzyme was synthesized. A new enzyme was not synthesized. How do we know that? It's because in the third finding, we find that the rate is constant, right? The rate wouldn't be constant if new enzyme was added, right? If new enzyme was synthesized, then the rate would have increased. Okay, so that's question 10. Let's go to question 12. All right, question 12. Let's see here. Oh, the same passage. I like this passage. All right, let's check it out. Extremely high temperature would have which of the following effects on the second trial? Basically, the second trial, remember we said the enzyme. Oh, I have food over here. I'm sorry, I need to eat. All right. Oh, okay. Jesus, basically. It's so good. All right, in the second trial, we had the enzyme, right? What does temperature do to enzymes again? If you have extremely high temperature, it's going to denature your enzyme, right? Enzymes work optimally in different conditions, right? Like for example, you have the enzyme pepsin in your stomach. That thing works at a really low pH. It's really acidic. Some enzymes can't handle that, right? Pepsin is unique. It can handle that pH. Some enzymes work, for example, in the human body, our temperatures are 98.6, right? That means most of our enzymes can work under that condition. However, if we had a different animal, let's say the animal lives in the Arctic. And if it lives in the Arctic, then its body temperature might be different. Maybe it's hotter in order to keep it warm, or maybe it's colder because of the cold environment. I don't know. But that enzyme functions at a different temperature, right? So each enzyme, even within the body, has its own optimal condition. If for temperature, if we were to decrease the temperature, under that optimal thing, then what's going to happen is that it's going to be colder, and the reaction is going to proceed slower, right? Remember, temperature is just like what? Energy, right? Temperature is energy, a measure of energy, heat. If you have a lot of energy in your environment, right? If you have a really hot environment, well, guess what? The activation energy that's required for enzymes to function. Remember, the reaction is this, right? This is your activation energy right here. This difference, right? This is the activation energy. What enzymes do is that they decrease. Let me just try it right here. They decrease that activation energy. So if you have if you're working under optimal conditions as an enzyme, then the energy from your environment is going to help you decrease that activation energy. However, if it's really cold, then you don't have the energy from the environment to help you kind of like decrease the activation energy. So the reaction is going to occur slower, right? Because the once decreased activation energy right here at the optimal, the less optimal would be like over here, like this dotted line, right here, this dotted line. This is what happens when it's really cold. Less activation energy discounted from the experiment, from the reaction. That's what happens during colder temperature. However, if you have high temperature, right, the enzyme is what? Is protein, right? Proteins denature at really high temperatures, right? So the rather than decrease the rate, the reaction is actually just not going to happen, because what is denaturing means? It means they change the structure. You're kind of unfolding the protein. And what gives the protein its function? Its structure, right? So at high temperatures, the correct answer, it would increase the required amount of potassium permanganate to approximately 28 milliliters. Why would it do that? Well, because the catalase that was once in there kind of got destroyed. So none of that catalase is doing any work. Remember, in the first finding, we found that 28 milliliters are necessary to kind of titrate all of the hydrogen peroxide. If in the second experiment the catalase were knocked out, well, guess what? You're going to have to use that 28 milliliters to what's it called? To take care of all the hydrogen peroxide, because catalase is not doing jack, right? Got the nature. Look at the other answer choices. It would increase the required amount of potassium permanganate to 18. Why 18? Well, it would make sense if it was 18 if it was a colder temperature, but in hot temperature, your enzyme is not even working. So it actually, you would need all 28. And C is what would happen if your enzyme was acting normally, because remember it said that it needed like about a third the amount of potassium permanganate. 9 is about a third. So that's like normal stuff. And it would reduce the required amount of potassium permanganate to approximately 1 milliliter. That's just not going to happen. The enzyme is not going to react better. It's not going to be better than it once was if it's beyond its optimal temperature. Okay, so that's question 12. Let's look at 25. Okay. Here we have okay, we have here a highly proliferating cell would most likely. Okay. Well, a highly proliferating cell. This kind of sounds like something that's really like maybe in your epithelium and stuff. Let's get some context from the passage, right? Okay, let's read. Okay, malignant transformation is caused by mutation and altered gene expression of oncogenes and tumor suppressors. Okay, do we need to know that for our question of a highly proliferating cell would most likely? Maybe not. Okay, so let's keep moving. These mutations can have thousands of effects on cell biology. Oh, snap. Okay, including affecting cell metabolism. Okay, work by blah, blah, blah. In the 20th century, discovered the Warburg effect. Tumor cells generate ATP mainly by glycolysis and lactic acid fermentation in the cytosol. Remember, what is glycolysis? Glycolysis is the anaerobic, means you don't need oxygen. What it is that you take your glucose and you oxidize the fuck out of it and you get 2-pyruvate, right? Glucose is 6 carbons, pyruvate is 3 carbons. So you basically split the glucose into two 3-carbon molecules, and in that process you make 2 ATP, right? So that's glycolysis. And what is fermentation? Well, basically during this process you need something called NAD plus. And what fermentation does, basically you need NAD plus for glycolysis, and you produce NADH. And what fermentation does, it just kind of regenerates your NAD plus, right? It takes the hydrogen from the NAD H and creates NAD plus so that glycolysis can keep going. That's what fermentation does, right? Lactic acid fermentation cytosol, the non-oxidative breakdown of glucose occurs even when there is sufficient oxygen available. Oh, so even when there's oxygen available, cancer cells just want to do glycolysis and fermentation. Okay, indeed, rapidly growing. Okay, and if you have a lot of fermentation, guess what? You can have a lot of lactic acid, right? Indeed, rapidly growing, because that's a product of fermentation. You take the pyruvate, you fucking reduce it, and it turns to lactic acid. Lactic acid is not good for your body. Too much of it. So indeed, rapidly growing malignant cells have glycolytic rates up to 200 times higher than those of their normal neighbors. So it's like, damn, they're really doing glycolysis OD. So high amounts of lactate are therefore produced in the tumor cells. Most of this lactate is exported. How do you export things from a cell? You shoot it out, right? Sometimes you do it in the vesicle, sometimes you do it in a protein, right? Okay. In addition to using glucose, tumor cells also can import. So import means to like get into the cell. How do you do that? With either vesicle or protein. Large amounts of glutamine. Glutamine is one of your amino acids. It's kind of like glutamic acid, except the OH is replaced by an amino group. So it'd be like your backbone. Your backbone's here. So glutamic acid looks like this, for example, and then glutamine would just look like, instead of the OH, it'd be, instead of the OH, it would just be the NH3, right? That's glutamine, which is useful for protein synthesis. Okay, why is glutamine useful for protein synthesis? Well, basically glutamine kind of triggers the cell to create more proteins, that's all. And also as the energy resource, right? Because we discussed when we were talking about protein catabolism that the amino group from an amino acid can be removed. And when you do that, the structure can be integrated into like the Krebs cycle or to the glycolysis cycle, or whatever. Yeah, so it can like, so you can get energy from amino acids by deaminating them, taking out the amino group and putting them into different cycles so they can do their thing, right? Glutamine lysis, the metabolism of glutamine to lactate is a pathway that distinguishes tumor cells from other normal cells. Okay, blah, blah, blah. All right, so let's check out this question. A highly proliferating cell would most likely, okay, it says over here, overexpress hexokinase and fructose 1,6-bisphosphatase. Okay, hexokinase is what? It's the first enzyme of glycolysis. So, okay, yeah, I mean, we said that the highly proliferating cell, aka the tumor cell, has like 200 times the amount of glycolysis than any other one. So yeah, they would probably most likely overexpress hexokinase. But wait a second, what's fructose 1,6-bisphosphatase? That is an enzyme involved in gluconeogenesis. Gluconeogenesis is the production of glucose. Glycolysis is the destruction of glucose. So you're not going to have two kind of cycles that does the opposite things, right? So this one is half right, half wrong. Look at C. Power cell growth by running the electron transport chain and oxidative phosphorylation? No, because remember, only glycolysis, only fermentation. D. Upregulate pyruvate dehydrogenase kinase and downregulate phosphofructokinase. Wait a second. Upregulating pyruvate dehydrogenase kinase, that's something that is involved with what's it called? The gluconeogenesis. Not really that important. And then phosphofructokinase is something that is involved with what's it called? With glycolysis, right? So why would you downregulate something that's involved with glycolysis? I'm sorry, I just need to like stand up real quick. My back is killing me. So basically, the downregulation of phosphofructokinase, that's one of the enzymes involved in glycolysis. That's not going to happen, right? So it's not going to downregulate it. That is, D is wrong. And then it says here, the expression expresses high levels of lactate transporter in the gluten. Is express high levels of lactate transporter and glutamine transporter. Yes, that makes sense. Why? Because it says that they export lactate and they import glutamine. That means they're going to have some sort of mechanism to do that. That transporter would be that mechanism. Okay, that's it for question 25. 3 more questions left, and then sweet, sweet Chinese food. Here we come. All right, 17. I'm going to the gym today too, so eating Chinese wouldn't be that good, but I'm literally like eating my own stomach. Like my stomach's digesting itself. Okay, 29. Okay, here we have a question. It has been suggested that one reason tumor cells have, oh, is it the same thing? Is it with the it is. Oh, nice. Cool. Okay, so it has been suggested that one reason tumor cells have novel metabolism is to generate the additional biomolecules required to increase biomass. What's biomass? It's just the amount of cells you have. What does it need? What does a cell need? It needs nucleotides to make its fucking DNA and RNA. It needs proteins for its structure. It needs lipids for its membrane, which is needed to support high rate of proliferation. Which is the best explanation for of how this occurs for tumor cells? Remember, what does a tumor cell do? It does glycolysis and also does what? Increase the import of glutamine. Okay, so knowing that, let's go into the answer choice. A, fructose 6 phosphate is shuttled from glycolysis to pentose 6 pentose phosphate pathway to support the both nucleotide and fatty acid biosynthesis. So, how does it support fatty acid biosynthesis? The pentose phosphate pathway. Well, the NADPH that's produced in pentose phosphate pathway is useful for fatty acid metabolism. However, it's not fructose 6 phosphate that's shuttled in pentose phosphate pathway. It's actually glucose 6 phosphate. So I got that one wrong too. So that one's wrong. High glycolysis rates can support the pentose phosphate pathway, which generates ribose 2 phosphate for nucleotide catabolism. Again, you're not going to be catalyzing nucleotides for pentose phosphate pathway. You're actually producing ribose 5 phosphate. That would be good for what's it called? For the production of the anabolism of nucleic acids, not the catabolism of nucleic acids. Okay, for D, glutamate is converted into glutamine to provide the cell with amino acid precursor, thus powering translation. Again, in the passage it says glutamine to glutamate, so this one has it reversed, leaving B. Glutamate can be used to produce the Krebs cycle. So glutamine is transformed into glutamate due to deamination, and then glutamate can be used to produce Krebs cycle intermediates such as oxaloacetate and citrate, and these molecules can be used to generate lipids. Okay, how does it generate lipids? Well, it generates lipids by producing acetyl-CoA, and then the acetyl-CoA or the oxaloacetate and the citrate, they're going to be used to generate gluconeogenesis, and the gluconeogenesis would be useful for the production of glucose 6 phosphate, and the glucose 6 phosphate was going to produce the NADPH, which is necessary for generating lipids. The amino acids, glutamate is an amino acid in and of itself, and glutamine is something that can generate amino acid synthesis. And again, the gluconeogenesis, the production of glucose 6 phosphate going into the pentose phosphate pathway can produce ribose 5 phosphate, which is useful for nucleotides. And that's why B would be correct. It's a huge mesh of a bunch of the pathways that you've learned before. Okay, second to last question, 31. Okay, let's go into it. Okay, here we have, okay, so it says here, as described in the passage, which of the following best describes how P blah, 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 blah causes disease formation? Okay, let's go look for it. Okay, it's here in the first paragraph. Awesome, let's see, let's look at what it does. In addition to viruses, other two non-living pathogens, pyrons and viroids, are transmissible in humans. Pyrons are unique in that they lack nucleic acids and compose completely entirely of misfolded proteins. Prions proteins exist in two conformations, one that is normally folded. Okay, so prion protein normally folded, so C, and one that is abnormally folded, so prion protein lacking formation is just SC, so misfolded. When the misfolded thing prion arises in a healthy individual, these prions serve as a template to induce normal brain proteins, so the things that are normally folded, to actually misfold and create more misfolding. So basically, prion causes a normally formed folded protein to become misfolded. So while the amino acid sequence of the misfolded protein remains the same as the folded protein, the misfolded protein causes the folded protein to misfold into tightly packed beta sheets, forming a structure called an amyloid plaque. These amyloid structures can interact with each other, causing the formation of a long insoluble chain of proteins. Amyloid plaques, I believe, are the things that are involved with Alzheimer's, and it causes deterioration of the brain. Here it says, to complicate matters, this conversion of alpha helices to beta sheets prevents the prions from fitting into protease activity, making prion proteins resistant to degradation. Oh, so that means that on top of making the amyloid plaque, the prions are not easily destroyed by proteases. Okay, let's look at the answer choice. As described in the passage, which of the following best describes how the prions cause disease formation? Okay, so look at B. Prions induce change in the primary structure. Nope, it doesn't. It says in the passage that it doesn't change the primary structure, so that's wrong. Prions induce change in the primary structure by, no, it doesn't change the primary structure. Prions induce changes in the secondary structure of normally folded protein by causing the formation of mutations in chaperone proteins, preventing proper protein folding. No, nothing mentioned about chaperone proteins. Okay, let's look at A. Prions induce changes in the secondary structure. Yes, because it's just misfolding, right? Folding is caused by what? Secondary structure, tertiary structure. Caused by alpha helices converted to beta sheets. Yes, that's stated in the passage. It says right here that to complicate matters, this conversion of alpha helices to beta sheets. That means that we're converting from one to the other. And that's what A is saying. That's question 31. Okay, our last question for the day. It would be the question number 48. So let's go to it. Here we have a fish probe. Okay, so basically you just need to know what a fish probe is. A fish probe is a structure that is going to act to find a specific nucleic acid sequence. So let's say you have a gene and you want to like find a gene within a sample, right? So a gene is typically, a double-stranded nucleic acid. If you denature that and you only get a single strand, well, our probe is going to be complementary to your single strand gene, and it's going to have a probe attached to it. So a probe. And so when the fish our probe binds to the to its complementary gene of interest, the probe is going to let us know that that basically the bonding happened. And if the bonding happens, that means that within our mixture, we actually have the gene of interest that we're looking for. Okay, so a fish probe should be made of, and C is the correct answer, a segment of single-stranded DNA, which is complementary to the gene being studied, covalently linked to a fluorochrome. And the fluorochrome is the probe, the thing that shoots out the light, so that people know that the probe has successfully attached to the gene of interest. Okay, let's look at A. An antibody, no, it's not an antibody. An antibody is a protein. We're looking for nucleic acids. A segment of double-stranded DNA, it's not double-stranded, it's single-stranded, so you can base pair. And a single-stranded piece of RNA, it's not RNA, it's DNA. Okay, that is it for the less peasy / not peasy sections. The video for the easy peasy sections will be posted up later this evening.